I hope I'm audible to all of you. Right. So this webinar is actually a shortcut for the 5 days, what you call hands-on training program, which we were supposed to conduct in the month of April. But because of the lockdown and then the COVID-19 pandemic, so we resorted to the next best option, which is a webinar for 2 days. And here I would like to invite all the three speakers, eminent speakers and scientists from Australia, CSIRO, Dr. VVSR Gupta from CSIRO Adelaide, Dr. Paul from CSIRO Sydney, and Dr. Andrew from CSIRO Hobart. So we welcome all the three of you. And today's webinar is on bioinformatic analysis on soil microbial community sequence data. We are all aware, and I'm sure this is specifically for the young researchers who have either started their careers as scientific officers or are doing their research as PhD programs. The new in vogue research area is plant microbiome, and how it is an important area and how it plays an important role in the overall sustainability and also in improving the crop productivity. So these three speakers will be taking one lecture of 1 hour each, and then of course followed by the discussion session. And without now taking much of time, I would like Dr. Viswanathan, who is the PI for this NHP-CAAST program, to kindly speak a few lines so that the speaker specifically will be getting the idea of what this NHP-CAAST program is, and also how we are going around taking different webinars and training programs with the specific aim to inculcate a scientific research aptitude, specifically in genomics, for the young researchers in India. So, Dr. Viswanathan, kindly, I invite you to speak a few things about NHP-CAAST.
Thank you, madam. Indeed, we are thankful to you for organizing this wonderful webinar from eminent scientists who are from CSIRO. I also, on behalf of the NHP-CAAST project and on behalf of the director IRI, I welcome Dr. VVSR Gupta, Dr. Andrew Bissett, and Dr. Paul Green. Phil, for this webinar. Briefly, I will tell about this program. So this is World Bank sponsored National Agriculture Higher Education Program. With the main mandate of this program is to skill the students with latest technologies so that they can do the basic and strategic research. So this program specifically at IRI is on Center for Advanced Agriculture Science and Technology. And the title of our project is on genomics assisted crop improvement and management. So this program is sponsored by Indian Council of Agriculture Research. And this program is headed by Dr. Arshi Agarwal. He is the national director. And then we have Dr. Prabhat Kumar as national coordinator. Earlier, Dr. P.K. Ghosh was looking after it. So these people constantly guide us on organizing various training programs. So our project, the main objective of our project is to work on genomics for the crop improvement as well as the crop management. So this project mainly aims at development of central facility for all the students of IRI and other universities who are willing to come and work at IRI, a common facility consisting of various molecular biology tools and facilities. And then the second aspect is to train IRI students on the aspect of genomics. So far, we have given about 90 student research grant. As we know that genomic research is highly resource intensive, so we need a special grant to the students so that they can carry out genomics work. So about 90 students have been supported from the research grant given by this project. And besides this, we have sent almost 19 students to get short-term training from laboratories abroad. So under this program, about 19 students went abroad, and about 17 students they have come back. 2 students are still abroad due to COVID pandemic. They could not get flight and return, and these students have carried out some part of the work. 2 to 3 months training they underwent in the international laboratories, and they could carry out some part of the research work there. And besides skilling the students, we are also supporting the young scientists who are willing to take training abroad in important areas related to genomics. So unfortunately, many scientists could not go abroad because of this COVID pandemic. Many scientists, they have got consent from the foreign laboratories, but they could not go. We hope the problem is ending, and at least some of them can visit before the project is over. Besides training the IRI students and IRI faculty, the objective of the project is also to train the faculty and students from the National Agriculture Research and Education System of India. So towards this, we have conducted about 11 training programs with the duration of 10 to 15 days each. And in this training program, almost half of the day practical and half of the day lectures were conducted, and all the lectures were recorded, and these videos are available in our website, so that the students can refer back to these e-resources to get their doubts cleared. And in this training programs, we have almost about 300 different students from almost all the states, that is 24 states, 3 union territories. The students have come and benefited from these training programs. Besides this training program, we also organized 2 workshops earlier, one on the rhizosphere engineering, a workshop which is organized by Dr. Annapurna, and another workshop we organized in collaboration with Colorado University on the computational biology for genomics. So in this workshop also large number of students participated and benefited. So this is in brief about the program. This year for the participating students, this year we are unable to organize any training wherein we physically bring you here and give hands-on training. So we started organizing the webinar so that at least some skills we can impart to the students at a large. So with this brief word, I thank Dr. Annapurna and Dr. Kumar for organizing this webinar, and I also thank the distinguished resource persons from CSIRO for kindly agreeing to deliver their lectures and teach our students the bioinformatics skills. Thank you.
Thank you, Dr. Vishnathan. I think we can now start the first talk in this webinar session. I would request all the attendees and everybody to mute yourself and also accept the speaker. All the rest can mute yourselves as well as go out of the video except the panelists. And any questions to be asked may kindly be put in the Q&A. And after the talk is over, the questions will be taken up and answered by the speaker or discussed by the panelists. I think that's the mode of things, Dr. Burman, right? So is that okay, Burman?
Yes, madam.
Yeah. So we now take the first talk by Dr. Gupta, and this is on soil and plant microbiomes in agricultural systems. Dr. Gupta has been an avid researcher on the microbial diversity. He's from CSIRO Adelaide. He had his PhD from Canada, and his interests are impact of agrochemicals and genetically modified plants on the functional diversity of microorganisms and soil biological health, and also on microbial ecology of managed subsurface aquifer waters. He's been a very active researcher in Australian systems, and I think Gupta is also in many committees, especially for the Australian microbiome research project which they are having. So without much taking any more time, I would like to invite Dr. Gupta. I myself have had very personal contacts with him and had visited his laboratory. He has been a source of inspiration to one of my own laboratory researchers also. Thank you, Gupta, for agreeing to be here with us for the next 2 days. I know that it starts late in the afternoon in Australia, but we are very thankful for all the three of the speakers. I would now like to invite Dr. Gupta to start his talk.
Uh, Thank you very much, Annapurna. Can you see my screen?
Yes, I can see your screen.
You have to go for a
Yeah, I will. I have clicked the button. I'm just waiting for it to respond.
slideshow we need, sir.
Yeah, I will.
It's coming. Yeah.
Okay. Well, first I'd like to say thank you to the CSRO team to be part of this. And thank you to Annapurna and Dr. Kumar and others, and especially the NIH program to facilitate this workshop. And I also express appreciation to CSRO for us allowing to extend what we know, what we do to all of you. I'm trying to change some of the things in my screen, so sometime I may take a break. Yeah. So the talk would be about soil and plant microbiomes, and with examples from agricultural systems. We all know that the productive and sustainable agriculture requires healthy soils and healthy biomes. And the new understanding from the new technology and associated developments in concepts has initiated a new era in microbiology research. So the talk, the general structure of the talk will be in 3 or 4 subsets. Under the presumption is soil biology plays a critical role in all aspects of plant growth, health, nutrition, input use efficiency. And this occurs even with elite germplasm. That elite germplasm on its own may not provide full potential yield, but requires a healthy relationship with soil biota. And we need this because, as we all know, the growing demand for increased production with the increasing world population. Biota in soils are diverse. They are diverse both in terms of their size, composition, and they are diverse in terms of their functional capacity. Many organisms are multifunctional, and they help plants withstand adverse conditions, acquire nutrients, protect against pathogens. So much of my talk will deal with microorganisms, which are small in size, but make up in numbers. And it is these microscopic organisms that are primary inducers of many soil-based functions. And the multifunctionality and resilience comes from diversity. And this is omics era. It's been for the last 10 to 20 years, and it is in its peak stage. We can now unravel the black box of microbial communities, and we can unravel the genetic diversity of this unseen world in our soils. The omics tools give us the power to understand the breadth and the depth of the microbial communities and its intricate composition, both at the organismal level and also at a molecular level. And this understanding also helps us to identify critical interactions of the organisms with environment. So it gives us power to know who is there, how management can influence them and modify them, and how we could modify it in a desired way. So I use the term metagenomics in this case as the definition given in the NRC report, that metagenomics is the study of microbial communities in order to understand them at the aggregate or a community level, that is transcending from an individual organism level and to focus on the genes and genetic potential within the community. And for this talk purpose, microbial ecology refers to the principles governing organisms, that is, interrelationships and interactions between microbial groups and their living and non-living environmental components. In the last decade, metagenomics has become a core of new age microbial ecology. It does have many phases determined by the methods that are employed and the complexity of the methodology and experimental approach, and of course, resource available. Number of techniques involving genomics are expensive to be able to apply to a variety of experiments. But what is also identified, the new research has identified that there is a need for creative ways to interrogate. I'm using the word creative in liberal sense, to interrogate the data and to actually uncover the intricate knowledge about the composition of the organisms and their functional capacity. But metagenomics only provides a predictive tool, a predictive picture of community functional potential. Metagenomics linked to metabolomics and proteomics and other functional related techniques would actually provide both. So, as I said before, the modern omics tools, there are a plethora of them. And these things go from a simple techniques as a profiling techniques, amplicon-based specific community techniques, to detailed understanding of their potential functions, that is transcriptomics and targeted metabolomics and proteomics. These different techniques allow to identify the structure of the communities in terms of the taxonomic composition and also their functional potential. This in-depth understanding needs to be linked to the processes, especially biotic processes, nutrient fluxes, balance between beneficial and deleterious organisms, in order to be able to link to plant responses. Because of the complexity of the system, both in terms of the composition and the habitat, a functional microbial ecology approach is required to distill the complex and detailed information. That is, detailed information in terms of a phenotypic diversity or functional diversity and the habitat characteristics, to be able to identify the links and the driving factors of function. And now the new developments in culture-based methods like the iChip and rhizochip technology can complement or integrate with the genomic tools to be able to see the unseen or unculturable. So that is an exciting development with this. There are generally two different approaches that are used. One is the direct extraction of the nucleic acids from the soil environment and then interrogating the nucleic acids, DNA, RNA, or the other way is to separate the cells from the matrix and then follow similar things. Both of them have their positives and limitations. And with both these approaches, the power of metagenomic tools can only be fully harnessed when all the principles and procedures required to interrogate a microbial community, and that is a complex microbial community, and the ecosystem function are rigorously applied. That is, the rigorous experimental design to account for habitat characteristics and the sampling procedures to acquire representative sample, to account for the spatial and temporal heterogeneity within the soil systems. In addition, the dynamic nature of the microbial communities, sample processing, nucleic acid extraction, identification of specific techniques for a community of interest, all of these things through primer selectioning and sequencing approach, all of these things need to be carefully applied to acquire relevant and reliable data, which needs to be later extended to application of the variety of bioinformatic and statistical tools. And my two co-speakers from CSIRO will address a number of these things. So I kept talking about heterogeneity. Soil microbiome is a composite of communities located in complex subhabitats or microsites or niches like aggregates, rhizosphere, mycosphere, soil pores, organic matter coating. Variety of these microsites hold distinct physicochemical properties, thereby allowing, encouraging specific members of the communities to flourish and function. And this complexity in both physical habitat and the community is a reason why soil microbial communities are generally stable, they're extensive, they're also protected, and it's been shown many people that they're also ancient. And the complexity extends from a general soil to roots and rhizosphere and cropping fields, because the complexity extends to the plant-soil interface below ground because of the distinct microsites and habitats, rhizosphere, rhizoplane, endosphere, they all have their distinct characteristics. And plants' ability to instill a selective enrichment of specific members through variety of signaling and host interaction pathways. So this complexity and diversity is good for managing a resilient community, but it actually makes it that when you're interrogating these systems, you need to account for the heterogeneity when you're sampling it. So we know much of these DNA-based tools use kits that use a small amount of sample, 0.25, 0.5 grams of sample, that is quite commonly used. But if you look at fungal communities or bacterial communities in a soil, whether it's in Australia or in India, anyway, where you have roots and no roots and stubble and pores, there is a variety of communities that are present at different sites, either at a micro scale or a centimeter level, or at a meter scale or a larger scale. So microbial populations are preferentially colonized in these microhabitats. So when we evaluated different sample sizes from 0.25 grams to up to 100 grams and looked at the composition and diversity of bacteria and fungi, in this case, example of fungi, that this different sample size indicated that more abundant OTUs were generally located in the hotspots, roots and decomposing residues, that is the patchiness. And therefore, more abundant OTUs are more likely to exhibit these significant differences among extraction sizes. Smaller extraction sizes seems to be able to access that locally abundant, but sometimes spatially rare communities. So you need to give attention to the system you are interrogating and select the sample size that gives representative picture of the community. The other aspect of this is how diverse is a microbiome, both at the organismal level or at a gene level. Because the organismal diversity, even look at the traditional ways, in a low organic matter, poor fertility soils may be less compared to organismal diversity in a rich soil, high organic matter soil, in the presence of a host plant. So the extent of sequencing effort required to get a full coverage of the sequence diversity in different environments can vary. So in this case, this is a graph borrowed from the work from USA, is that the sequencing effort required for an agricultural systems, in this case soil systems, to get a full higher coverage or better coverage is different to that from a human microbiome. Some pictures are missing here in the beginning, gut microbiome or water systems, and these are for soil systems. So what they have found is that for an Iowa soil, you require 0.6 to 1 terabyte of sequencing effort to get a 95% coverage of this. And this applies for both metagenomic and transcriptome approach. This is more critical when you are looking at specific groups of organisms. So in this example, I'm giving the number of sequencing reads or sequences you can get for a functional gene involved in nitrogen cycling, that is NHP, versus a carbon cycling gene, COX-L. And you can see for a variety of these environments, from arable soils to deserts to prairies to rice paddies, with a decent amount of sequencing effort, the number of NHP sequences are quite low. So if your aim is to understand the composition of the NHP sequences with this level of sequencing effort, you may not get enough depth of coverage to be able to do. So another point you to look at when you're looking, especially for a metagenomic work, not an amplicon enrichment-based work. So in this scenario, the depth of sequencing effort has to be given a critical consideration. I think Andrew may also cover a bit of this. The next level is at what level you are looking at the microbial diversity. Because the drivers of microbiome, that is microbial communities, occur at different scales. Whether it is a micro scale, I talked about roots and plants or plant species, or at a macro scale, at a regional level, or a continental landscapes. And because of these drivers that control the composition, abundance, function, capacity at different levels vary. So in this case, we have given a hypothetical 0 to 5 scale for the relative importance of different drivers, that is energy related stresses and host presence. You can see that the ones that are 5 means they are more important. If you're looking for a regional variations in communities, then you need to know many of these habitat character properties to be known. But if you're looking at a plot-based or experimental plot-based, the factors you require are quite different. Information you require is different. So you need to understand there is a need to understand the community structure and the responses to the various driving factors in order to identify the stimuli for a specific group of communities, either host impacts or management effects. So I'll go now to the examples, that is the examples to interrogate microbiome. And I will give examples, 2 examples for diversity at different geographic scales using a taxonomic group, Actinobacteria, or a functional group, diazotrophs. I also use a couple of examples related to biological disease suppression of plant diseases or host genetics. In order to describe or present the how omics tools are used for these different examples, you need to know that the exploring omics data in terms of the compositional variation or responses to stimuli, there are a variety of tools available. To interrogate the omics data in terms of a composition, community structure, and variation. And some of them are traditional abundance-based ones, are diversity-based measures, variety of species diversity, richness, or phylogenetic diversity. These are all you would have seen alpha diversity measures. Or beta diversity measures, that is an interrogation of a similarity or dissimilarity between the factors or the treatments that you're looking at. So I will use examples of results we found for those 4 different categories using analyses results using these different types of analyses. How to do that? Many of you know there are a lot of online pipelines and other procedures to conduct these analyses. So the first example is where we looked at the distribution of Actinobacteria. Most of you would know that Actinobacteria are one of the important groups of soil microbial communities, both for their abilities to provide human health benefit functions, but also plant health related functions. For this, we used data from Biomes of Australia Soil Environments program that has been there in Australia for the last 10 years, for which Andrew Bissett is a scientific coordinator. We utilize the data that is generated from this base program using base protocols for samples that 974 sites across Australia, mainland Australia, and at more than 250 of the northern Antarctica samples. And the samples, as you can see, are distributed across Australia, different regions. We use the 16S sequence data from this, and we use the metadata, physicochemical properties, again, provided by these labs and also acquired by this base program. So what did we find? So what we found is that many Australian soils are known to be poor organic matter and lower rainfall, lower resource input systems. But in these environments, what we found is there is a rich diversity of Actinobacteria. And the proportion of Actinobacteria as a proportion of total bacteria can range from around 5% to 30–40%. And there's a more than 6,800 OTUs, and more than 2/3 of them are rare OTUs present in some environments. And what we also found is that members belong to the 5 families accounted for 70% of the composition. So that means 24 OTUs were more frequent, considered as frequent, like more than 5 or 10% compared to 2/3 of them rare OTUs. What we also found is the extreme environments like northern Antarctica or central Australian deserts showed high number of these rare or endemic OTUs. So rich diversity, but few of them are enriched in some places. When we looked at the similarity of these composition communities in different regional based environments, there is a significant beta diversity based separation between Antarctica soils. So you can see colors from one side to other, green to the brown. They go in a systematic fashion. And this dissimilarity is driven by 10 variables related, like latitude, pH, variety of these things. And this dissimilarity is associated with variations in a few members of the community. You could say responsive communities. But actually alpha diversity measure across, in this case, Shannon diversity, is quite similar across different environments, except for these rare Antarctica and Christmas Island. One of the things that we looked at is that, is there an effect of disturbance, in this case, mining, agriculture, urbanization? So on this graph, what we are looking for is the disturbed sites. These are all these darker color sites. The disturbed sites to be more closely related than the undisturbed sites from different parts of the country. Undisturbed sites from national parks, conserved areas, and nature conservation areas. So it's showing that the disturbance led to more reduced variation in the diversity, which you can see in terms of these dispersion indices and similarities. So disturbance reduce the diversity in a way that few of them are enriched. So that's about a taxonomic community. The second one is the a functional group. Another example. In this case, I'm using diazotrophs and data from a amplicon sequencing using the functional gene NIFH. And in this case, we only are looking at what is the diversity and functional capacity of diazotrophs in wheat fields. So we remove that plant-based variation or influence. And we use soil types and systems from Eastern Australia to Western Australia. And there were more than 100 known genera, and from 25 to 45% were unidentified. And Proteobacteria were the most dominant, more than 50% of them. One of the significant factors that influence the diversity is the soil or habitat characteristics. We know diazotrophs require microaerophilic conditions and carbon-rich environments. So vertisols are clay soils that provide numerous such microsites showed high diversity, whereas sandy soils, where such communities can only flourish at decomposing residues and roots, a low diversity. So habitat controlled in this case at a continental level, which you can see when you look at the beta diversity data. So in this case, the Western Australian sandy soils community is quite distinct to Eastern Australia and the Queensland, New South Wales soils, which are more texturally clay related, compared to the Southern Australian environments of which have chemical constraints. And in this case, the factors that are separating these communities are habitat properties and of course rainfall and pH and things like that. So at a continental level in wheat fields, it is a habitat characteristics and environment that drove the composition. Whereas at a regional or a field scale, it's a management that drove, in this case, stubble management, fertilizer application. So depending upon at what level you are looking for the composition, the drivers that change diazotrophs are different. When you look at the communities that are complex but are influenced by a plethora of habitat properties, then microbial networks provide a spatial snapshot of community structure in a given environment, because they provide ability to integrate multiple types of information. So we use this random matrix theory-based network analysis procedure. And what we found is that the network, diazotroph network community structure between vertisols and sandy soils varied in terms of the community network structure as defined as the nodes and modules and linkages between nodes. And these differences are related to the soil properties. So majority of the correlation between the modules and habitats were seen significant in vertisol, because these soils have variety of microsites compared to the sandy soils. So interrogating omics-based community composition-based data, not just in terms of the similarity composition, but also in terms of relatedness between the properties, helps to provide another level of information for this. So from this work, what we found is relating this to the functional nitrogen fixation capacity using N15 assay, is that diversity is important for functional stability for this functional group. So the next example is about management influence on microbial communities and agricultural systems. And for this, I will use this biological disease suppression. So it refers to the ability of soil to suppress disease severity, even in the presence of the pathogen, in the presence of the host plant, and a favorable environmental conditions. So I'm using the example of Rhizoctonia bare patch disease caused by Rhizoctonia solani. And Rhizoctonia solani is a very capable saprophyte that persists and survives and takes advantage of a low organic matter nutrient availability systems, such as that's seen in many parts of Australia or in India. And what we found is that even in these low fertility soils, long-term practice of conservation agricultural practices, that is stubble retention, reduced tillage, led to the increased inputs of carbon, that is energy source for microbes, reduced disturbance, allowing microbes to evolve and develop, and resulting in a lack of disease after up to 7 to 10 years under different crop rotation scenarios. So it is the input of carbon energy source in these low fertility soils that led to the change of the community composition, led to a community that actually supported suppression of a disease caused by pathogens. So what was the hypothesis? That this high level of stable disease suppression activity against such pathogens is a product of phylogenetically and functionally diverse group of communities. you can't just go and isolate few stuff and interrogate such things like this, especially when the plant pathogen microbe interactions for such pathogens involves interactions both in the absence of a pathogen, that is non-crop season, and in the crop season. So it's a more than a decade of research, classical research, looking for individual organisms, biocontrol organisms, to control the disease. And a number of bacteria, fungi, actinomycetes, protozoans, nematodes, all have been isolated, found to reduce disease suppression, disease incidence, that is biocontrol, in a controlled environmental studies. But presence of this organism, addition of these organisms did not explain variations in a field environment, did not explain variation in disease occurrence in multiple seasons, and suggesting that a community-based role in this disease suppression. How can we look at the community-based approach? We use this comparative metagenomics and metatranscriptomic approach. And we use this with two different scenarios. That is, one, rain-fed agriculture in southern Australia, 3 different sites, where we found sites that are close to each other, within 500 meters from each other, that are seen to be suppressive and also conducive disease. And another example is in Eastern Australia, cotton growing country, again, fields that are known to be suppressive and conducive. We use this comparative analysis approach with sites with various suppression potential and what we call a targeted polyphasic approach. That means we use the sites as a factor, fields with a different suppression potential, and we also introduced a seasonal factor because it is known microbial communities can change depending upon the season. We also interrogated or tested the ability to regulate the expression of suppression. That is, by linking the microbial community diversity and functional capacity to a trait or a system trait. In this case, our hypothesis is that increased carbon inputs that led to the suppression development. So in brief, we used a functional microbiome approach. So we measured the microbiome composition. We tested the drivers that either promote suppression, suppressive microbiome, or restrict. We also looked at the factors that regulate the expression of suppression. So we used this is polyphasic approach. This is where we applied the metagenomic tools. And by taking into account the variety of factors and principles I indicated before, we used adequate replication to cover for the spatial heterogeneity. We determined this before by testing the samples and preliminary experiments. So how many replicates do you need to get a reproducible community composition data? We used both amplicon sequencing and direct metagenomics and transcriptomics. And we followed the various bioinformatic procedures, the things that you're going to hear from Paul and Andrew, and used various statistical other interrogative tools. So what I'm going to present is the outputs of all these analyses under 4 or 5 different points. The first one is metatranscriptomics. So we use metatranscriptomics of rhizosphere or root microbiome sampled at the peak of a disease incidence in a wheat fields. That is around 8 weeks after the wheat plant is germinated. That's the time it is known that the rhizoctonia disease incidence at its highest and broadest. So what did we find? When we looked at the transcriptome data, that suppressive microbiomes are distinct composition to a non-suppressive microbiome. Suppressive microbiome showed a dominant differentially expression of a taxa related to alpha and beta proteobacteria and members of Actinobacteria. So there is a distinct taxonomic or phylogenetic composition of suppressive communities compared to the non-suppressive communities at these 2 sites and 2 different fields and 2 different seasons. If you look at the transcriptomics data, based on functional categories. Again, you will see differences between suppressive and non-suppressive communities. They're simply based on blue colors, which are means red more dominant, blue less dominant. Suppressive microbiome showed greater expression of functional genes such as polycyte cyclase, terpenoid biosynthesis, and cold shock proteins. So that means that diverse functional communities are operating in the suppressive community. When you looked at that, transcriptomics gives a snapshot of at the time of measurement. But rhizoctonia disease or pathogen can infect, cause disease over a long periods of a wheat growing, from 2 weeks from germination to up to physiological maturity, over a 3-month period. We can go and do transcriptomics every week, changing things. So we said, okay, let's look at metagenomics, DNA-based analyses. And in this case, we used 2 different sites, a suppressive site and what we consider a less suppressive site. We used 2 different systems, that is systems with just pathogen or systems with enriched carbon inputs. When we look at simple diversity, Shannon diversity, you notice the response to factor, in this case carbon inputs, in a suppressive soil in terms of diversity is more than in a less suppressive soil. This is overall composition, metagenomic composition of the community. And there are some groups of in this metagenome community composition that are more responsive to this. Those involved in virulence, disease, and defense related ones, which you would expect with various levels of disease. Those that are involved in quorum sensing, of course, plants change selectively microbial communities, the root microbiome, through these sending these quorum sensing and other signal tools. Those that are involved in nitrogen metabolism and stress response to plants. So the transcriptomic approach showed both taxonomic and functional. Genomic approach showed that the transcriptomic results from a single point actually extend to the community composition when you look at like a genomics based things. So this is all looking at functional categories that way. Classical approach to see, are there bacteria, are there fungi that are different? So for that, we use the amplicon sequencing technique. So this is the data from a 28 LSU based amplicon sequencing techniques for fungal communities. So this beta diversity plot shows that there is a significant difference in fungal communities, that is fungal communities in a suppressive fields. In this case, there are 2 suppressive fields that are blue, there are 2 non-suppressive that are green. So there is a significant variation between based on suppression potential, and this difference is statistically significant. But there is also a difference based on locality. The A1 and Minapa are the 2 sites that are located around 600 kilometers apart. So different, slightly different rainfall patterns, some soil type based. So within the background of site-based variation that I said before that dictates the community composition, soils with higher suppression potential have a different community to non-suppressive soils, fields nearby. What is contributing to these variation? Suppressive communities are generally more diverse and richer. And non-suppressive communities had number of them are putative disease-causing organisms. So putative pathogen abundance is greater in a non-suppressive community. So these are those blue ones. It may be difficult to see individually, but that's what it is. Suppressive communities also have members with known abilities to fungicidal activity, endophytic activity, antifungal activity, all of these things. So while non-suppressive community is dominated by putative pathogens, suppressive communities is dominated by members. These are members from, like for example, hypocreales with known fungicidal antifungal activity. The interesting point, because we use multiple seasons, that majority, that is 14 out of 20 of these genera differences were similar both seasons. So suppressive fungal community composition seems to be stable across 2 different seasons. The second one is bacterial communities. So for this, we looked, because bacterial communities change with more readily with changing environmental conditions. So environmental conditions at seeding may be quite different to environmental conditions later in the season. So then we looked at what is the responsiveness of these communities to the addition of carbon, because our hypothesis, addition of a carbon over a period of time, either through management practices or root exudation, rise or deposition, leading to this development of suppression. So what we found is that suppressive soils showed higher diversity of bacterial communities. That is general, which actually means the wheat plants have more opportunities or more members to be able to choose the beneficial taxa. And the response of the bacterial communities to this carbon addition, we were able to group to 3 different types of responses. There are members that are always higher in the suppressive soils than non-suppressive soils. There are members that are more responsive in suppressive soils to carbon addition than non-suppressive soils. And there is a third group of members that are greater responsive in these non-suppressive soils, because higher redundancy in a lower organic matter soils led to identifying individual communities over a season is difficult. So we were able to see the communities that respond. And many of these communities have this chitinolytic or polychaete synthase. You may remember the transcriptomics data showed similar trends. What's interesting was that addition of carbon did not help suppression develop in the short term in a test in these constrained soils of alkaline calcareous soils. We didn't know why these soils weren't developing this suppression. By doing genomics, this analysis, we found that in these soils, these putative suppressive organisms like Actinobacteria and Actinobacteria, they didn't respond when you add carbon. So something is restricting the communities to change. As I said before, functions that are mediated by complex interactions require understanding the community structure. So again, we use this network analysis. What we found is that suppressive microbiomes seems to have more structured communities. That is, there are more number of nodes in the suppressive, both at sowing and in crop, than non-suppressive. More links means suppressive fungal communities are characterized by more resilient community structure, whereas non-suppressive communities, they varied with time. The structure lost as the crop grew, pathogen invaded. And in macroecology, it is known highly connected networks allow all species to be able to use resources similarly. And these interactions between communities also showed that putative suppressive microbes such as genera such as these, Cephalophora xylaria, have more linkages to the pathogens in a suppressive community. That means they're able to control pathogens' growth, ability to cause disease. And this dryland agriculture-based work findings were reproduced in irrigated cotton systems. I'm just showing one figure. You can see the more structured community in suppressive soil, better modularity and more diversity compared to a non-suppressive soils. So a complex biological function such as suppression could be explained by understanding the community composition in terms of a specific groups, bacteria, fungi, or in terms of their functional capacity. So the level of suppression activity against such pathogens is a product of phylogenetically and functionally diverse. So the last example is host genetics influence and rhizosphere microbiome. So I have around 10 minutes of talk. Is that okay, Annapurna?
Yeah, sure.
Okay. Okay. It is known from culture-based other works that there are plant-based variations in the communities that the plant species attracts, either in the roots, inside the roots, or in the rhizosphere. So the degree to which a plant genotype can affect the composition of associate microbiome, either general composition or diversity, evenness, or functional density, or specific functional groups. And there are examples in the literature for wheat, barley, cotton, maize, and peas. So plant-based variation is known and reasonably established. But which component of host genetics regulates these? There's a limited evidence for this for a couple of. I'm going to use an example where we looked at host genetics in terms of a wheat domestication and its potential influence on root microbiome. So this graph is an example of bacterial microbiome using amplicon sequencing, where we compared 3 different wheat varieties, 16 different land races of Triticum istivum, and 8 wild relatives of Triticum. And this beta diversity plot shows that the root bacteria microbiome composition is dissimilar, different from that of a land races and varieties. You can actually see that in terms of this Venn diagram base, that these individual groups of plant the types seem to attract and select members that are only attracted or enriched each of these plant types. But overall richness is not big difference between them. But what type of organisms attracted is different. How does it actually affect? By just showing that, yeah, different relatives and varieties differ. So is that difference is systematic, or is there some relationship between these variation to host genetic similarity? So from those 20 or 22 modern cultivars we tested, we took the data for 7 parents from CSRO breeding population, and for which we got information on the genome similarity between these 7 cultivars, and this is based on percent SNPs for between the cultivars. We looked for relationship between this genome similarity from these SNP-based genome similarity estimates and microbiome variation. This is microbiome variation for bacteria and fungi. And you can see that although the relationship is not stringent, but bacterial community variation between these cultivars is significantly related to the genome similarity of these cultivars. But you don't see that much with fungi. In a way, you could probably explain fungi can have hyphal networks extending from a plant root to outside. It is a hypothesis. So for a wheat one, we looked at control environment. There is a literature information for this for maize. So this is a work by Buffard et al. And you can see in this case, a similar graph for a phylogenetic distance versus rhizosphere community, quite strong evolutionary relatedness between the Poaceae family members and Rhizobacteria. This is even with a background variation that you can see is soil type based and a varietal based. So there is some host genetics based control on the microbiome. They actually attract and enrich. The final example is another one for host genetics. In this case, we're looking at diazotrophs associated with perennial grasses. Perennial grasses, different perennial grasses in southern Australia. And the presumption that plant species-based NIFH composition varies. So we used NIFH amplicon-based analyses, and simple relative abundance shows that there is variation between, in this case, we looked at below ground communities in the roots and above ground communities, shoots and leaves. And there is a diverse community both in below ground and above ground. Either you look at relative abundance or phylogenetic relatedness. Surprisingly, there is quite similar. This is Shannon index, both below ground and above ground. Our hypothesis is that above ground have a limited diversity. And between the plant types, there is a significant variation. These are different below ground and above ground components. But you also have a different plant part variation composition. So there's plant-based selection. So when you look at that, you look at community composition. So what is found is that individual plant networks, this is roots, stems, and leaves, have a larger, more complex communities independently. But you combine them, the community between plant types is similar in terms of the modes and nodes and the members involved with that. It suggests that there is co-occurrence patterns of the diazotrophs is compartmentalized between different parts. Each part of the plant represents a unique niche. You know the niche in the roots is different to niche in the stems, and the niche in the leaves is different. We are looking for a functional group of communities, diazotrophs, with specific habitat requirements. You know, microaerophilic anaerobic environments, high energy supply, low end product presence, that is nitrogen. So it is not surprising to see that these different host plant parts have a different communities. Are they selecting them for diazotrophy or something else? It's a bit more complex one, but a simple point is that the selection by different plant parts seems to be not from the presence of a NIF-H ability, but more related to other phylogenetically related property. And so because it is known that there is a functional overlap and redundancy in a functional group such as diazotrophs, that is fixing. This may also extend to other functional groups such as ACC deaminase producing organisms, you know, ACDS community. But it is actually known previously in rainforest plant species, where it was said that the stochastic processes dominate community assembly in different parts of a bacterial community. That is a perennial trees, but it seems to be in these perennial grasses, but their annual growth and non-growth habitat. Diazotrophy is more of a stochastic than deterministic for the functional ability. So some other factor controls the selection. What was surprising is that the communities above ground of the perennial grasses in Australia are quite similar to the communities in perennial grasses in USA. You can see the stems and leaves are quite similar, but roots, because roots are more influenced by soil. So a strong host genetics based selection of diazotrophs, not necessarily for diazotrophy, but for some other relationship. So what does all this mean? I hope that I've given a convincible argument that harnessing the power of microbes through the omics tools and associated experimental approaches provide more in-depth and useful information that can help interrogate community diversity, community functional capacity at a broad biogeographic distribution or regional or local management effects. And integration of omics tools with functional microbial approach in a system-based experimentation is critical to be able to distill useful information. And a final note, I say it's certainly an exciting times to be a microbiologist. And one of the persons that you may know, Albert Rivera, in a way, an eminent microbiologist from 1950s and 60s, what he made a comment is the opportunities for microbiome research is now are similar to when he went into rhizosphere microbiology in 1950s, when variety of biochemical techniques were incorporated or integrated with culturing techniques. So for younger people, take advantage of these new techniques and opportunities, apply them appropriately, and enjoy. Thank you very much.
Thank you, Dr. Gupta. An excellent talk. And I do hope all the youngsters listening to this talk will be greatly enthused to carry out the research, their research programs in this very novel and exciting field, and that is microbiome. And like we have read and Dr. Gupta's presentation, plant alone is not sufficient. It's not unique in the sense it's not isolated, and it is always and always associated with microbes. So that's from there itself, from a single isolate, the concept of a collection of microbes came into existence, that is microbiome. And then of course the techniques and tools to study the microbiome. So thank you very much. And I would now like to start the discussion part on this talk. I see there are quite a large number of questions appearing in the question answers, and I would ask Kumar to kindly take over.
Yes, ma'am. It was a wonderful talk, and Dr. Gupta has covered almost every topic. There are several interesting questions also. Some of the questions we already answered. I just pick up some of the very interesting questions from Dr. Murali.
Kumar.
From CPCRA Kasaragod. This question is to Dr. Gupta.
Mic is mute. You can.
Yeah, can I read the question, sir?
You can ask them to directly ask also few questions. I think that will be, they may be interested.
Yeah, that would be better. Yeah. Dr. Murali, you can ask the question directly to Dr. Gupta, please.
You have to permit them to ask the questions. You asked Dr. Burman to permit them so that they can ask the question. All the attendees, they can ask the question directly.
Actually, they are not permitted to ask questions, I think, directly.
Well, Vishnathan, if we ask the attendees to ask, I think it's better if one of the co-hosts asks, because then they are not with the systems with the proper net connectivity also. So I think.
Madam, are you talking about Dr. Murali Gopal?
Yeah.
You can permit, allow him so that he can ask some questions.
Yeah, he's allowed, sir.
Yeah. Dr. Murali, you can ask the question.
Good morning, everybody. Am I audible to everyone?
Yes.
Yes, you are.
Thank you very much, Dr. Annapurna Madam, Dr. Kumar and Dr. Vishnathan for this opportunity, and to Dr. Gupta for a very insightful talk. We are working on a very difficult crop, coconut, and it's a very perennial crop. It grows for more than, its life cycle is for more than 100 years. I would like to know, would this crop have a more stable and more settled core rhizosphere microbiome compared to the annuals? And second, would this core rhizosphere microbiome also be selective for the abundant OTUs, or it will have only the, it will have mostly the abundant OTUs, and would it not have the low OTU that comes up in the data? Thank you.
Get the question, Gupta.
Yeah, I did. Thank you very much. And so what I'm going to say would be based on my general perception than my experience with the coconuts. Perennial crops certainly have more likely to have a stable community once a plant is established. There would be some changes in the seasonality depending upon the crop. And from what I know of coconut, it's not like a deciduous plant. That I would see more stable community within the roots. There may be differences in the rhizosphere seasonally, but general community composition would be distinct for a coconut plant compared to something else. In relation to these abundant OTUs and rare OTUs, that depends upon how you are looking at, which component of the root or plant microbiome are looking at. Because plants selectively enriched some members. So there will be, of course, some members that are a lot more abundant and than in a general soil. And it is now well established that the diversity of microbiome in the general soil is actually more than the diversity near the root and inside the roots. But the selective enrichment is, of course, is greater within the roots. So if you look at richness and evenness and the type of alpha diversity, it'll be more in the soil than in the roots, whether it is coconuts or perennial grass or wheat. But if you look at relative abundance of selective groups, you will have, or even total populations, greater closer the root or inside the roots than in the bulk soil.
Thank you very much, Dr. Gupta, for the clarifications and your suggestions. Thank you very much. I'm able to get some idea. Thank you.
Yeah. One of the question is how to identify core microbiome in cotton when each year farmers change the variety or hybrids which are released.
Oh, there is a seasonal based variation in, if you look at a broad community composition. But now it has been shown for a number of plant types, including cotton, but there is a component of microbiome, root microbiome, that seems to stay with the cotton crop in spite of the seasonal variation between seasons and within a season. So what is a core microbiome? A component of microbiome that persists with that host plant or a soil type or a system.
The one which is tightly associated with the plant, we'll call that as a core microbiome.
In general, that's what it is associated. The one that is less variable, that makes it distinct from the outside environment.
One more question. This is by Kamlesh. The high degree of rare OTUs in your data is intriguing. Could that be due to a difference in the way the sequences were generated using different technologies as they were picked from a database?
My guess is that this is to do with actinobacterial data. The actinobacterial data comes from variety of environments. These are environments like desert soils in central Australia with a dry, hot environment, low carbon, very little plant presence, and cold, harsh environments of Antarctica, rich pasture soils of Eastern Australia, and in crop fields. Because of these diverse sample or systems that are interrogated, you have a likelihood of finding members that are specific to one environment or other that were present in lower abundance in specific systems only. So with actinobacteria, that would be one of the reasons. I'm able to say this is, you will also hear when Andrew talks about this and Paul, that it is possible techniques that you use can result in such observations. But in this case, the base program used all standardized, reliable techniques from sample collection to data interrogation. So analytical based observation contributing this rare OTUs is less likely than the system variation based difference. But it is very possible if you use multiple techniques, if you use not so thorough bioinformatics tools, all of these you will learn from Paul and Andrew's talks, how to avoid such things.
The other question is, at what level of occurrence do you define OTUs being rare or abundant?
I think it is also the actinobacterial one. So what I'm going to say is more from memory than exact number, but I think we looked at ones that are less than 5% were rare, less than 5% of the samples. And the abundant ones were the ones that were accounted for more than 5%, almost more than 10%. And because of the sample number, large number of sample number, we were able to take such strict categories or thresholds for identifying more abundant or rare. These categorization, you will find quite a big of variation depending upon system people are studying, sample set, type of analyses. You know, there are people who use 0.5% or less as a rare, more than 30% or everything. We chose this based on the data set for actinobacterial communities in the data set that we interrogated. So there is some loose categorization thresholds used for this categorization.
Okay, another question from Reddy, Mr. Reddy. What primer sets were used in the study for NIFH? And does this primer set cover paralogs? How to remove the paralogs from raw data?
Oh, there is a lot of papers on how to process NIFH or a functional gene-based data set. There are papers by few of them. I can put in there and you can have a look because it's from me going through this, the operational facts of that. But the primer sets there are two sets of primers that are quite commonly used. One are polyprimers, Poly et al. I think it is early 2010s. And the other one is Zer primers. And these are, again, they both, there's a paper by Buckley et al., I think in 2005 or 6, which compared various primers. Most of the primers, the coverage is between 35 to 50% only. That's one of the things with NIFH primers. But poly primers are considered as one of the ones that highest coverage within limitation and also covers diverse groups of them that are things that are spore forming anaerobics compared to our highly responsive proteobacteria. So polyprimers are the ones that are more commonly used. But the two reviews by Buckley et al. Certainly talk about, they evaluated the ability of different primers in terms of their coverage, reproducibility, and representation of the community that is present.
I think Dr. Ramaswamy has some query.
One more question. Both are on sampling only, ma'am.
Okay.
Yeah, in a 10,000 hectares area, how much sample we need to collect to have a representation of the entire area? It's a difficult question.
It's a difficult
and
question.
The question goes to even continental scale also.
Yeah. You see, you are looking at, do you want one number for the 10,000 square meters, or within that geographic area, are there distinct habitats in terms of the host plant, environment, soil type? You need to look at that. See, actually, you should look at the base protocols. Base protocols are, if you can type Australia microbiome, a link, you will find on Google, and they have protocols, how sampling protocols, how to identify various factors that needs to be considered in collection of samples and sampling procedures. So on a side note, when you're looking, farms in Australia are, 500 hectares plus, most of the farms. So when there is a diagnostic service in State Department in South Australia, that farmers can send samples to know the abundance of plant pathogens for wheat, potato, and peas, things like that. So their procedure in general tells farmers that you need to have at least 30 individual samples distributed across in a 1 hectare of a land. So if you have a field, wheat field that is of 20 hectares, you need to collect those individual samples of around almost 100 of them to get a representative sample for measuring soil plant-based plant pathogens. So that's where they actually looked at that. But a sequence-based approach, it depends upon that regional, local-based one.
I think Dr. Ramaswamy would like to ask.
Hello, Rajan.
Oh, sorry. Yeah. Are you there?
Yes, connect.
Thank you, thank you. I think he has answered my question on sampling. Thank you very much.
Asked the same question.
Okay. Okay. There's one question for, I think, which Andrew would like to answer. What is MIFECH amplicon sequencing and its importance?
Andrew, are you going to say something?
Yeah. Madam, I have a small query for Dr. Gupta. Can I ask?
Go ahead.
Madam, can I ask?
Yeah, ask.
Yeah, Gupta, it was a wonderful talk. And there was a lot of questions about core microbiome. And people are asking whether the abundant one is a core or rare one is the core. My question is whether the seed associated microbiome is the core. Because you also made a point that across generation, same association, same microbial community, which is associated with the plant, can that be called as a core microbiome? That means that seed is a connecting link between two different seasons. So can we say the seed microbiome as a core microbiome?
It's a very fluid terminology for core microbiome. It is generally used to refer to a community members that are associated with that genotype. And there is research that shows that seed microbiome can vary depending upon where the seed is coming from. So therefore, categorically, wheat seed microbiome is core microbiome is not necessarily, because again, if you look at wheat seeds coming from same variety of wheat seeds coming from Eastern Australia, Western Australia, India, Africa, and if the members of the community that are present in all of these wheat seeds, then they would be core microbiome for that wheat, variety. So it is more of the members that are more frequently associated with that host genotype, not necessarily in its abundance specifically.
Okay, you mean to say that core microbiome is something which is recruited during the crop season?
It is present with that crop specifically.
Okay. Yes, sir. Thank you.
There's one question from Mr. Hemant on the lighter side. How to get such beautiful graphs? Where has been used?
Oh, it depends upon different analyses, different things. So I'm sure, I think mine are not as good as many these days. There are a lot of tools, online tools.
I have a question for Dr. Gupta.
Yes, Ramakrishnan, go ahead.
Yeah, Dr. Gupta, thank you for an excellent talk. We learned a lot of things from you. I have an observation. You said organic carbon and nitrogen plays an important role in the community structure from a different scale, from continental to the field and plot. Did you find the application of nitrogen as having a serious or significant effect on actinobacterial diversity?
In that data set we looked at, there wasn't enough depth to interrogate just fertilizer effect alone. But
Okay.
there are other studies in the literature that management practices such as fertilizer application could have short-term changes in community composition. But it depends upon which systems, how often it is applied, how much is applied, all of those things. And also looking at whether you are looking at total abundance or composition of specific groups, all of these things. So in our study, we weren't able to interrogate specific nitrogen application effects.
In continuation to that, could these changes, if at all, with N application be very transient? Because as the N is utilized by the plant, the equilibrium is coming back again to the original.
I think our application of fertilizer is having a stronger effect because we are actually changing the diverse ecosystem because of application of fertilizers. So I thought probably you're seeing something like similar to these effects, like fertilizer application is actually reducing the diversity of communities and soils. Well, thank you.
It's not, for the question Annapurna said, the effects of these anthropogenic activities like fertilizer application, chemical, depends upon the level of disturbance it causes and the duration of the disturbance it causes. If you go and apply one time and have a look at it, you may see short-term changes in some abundance and composition. But once that stress or a factor is removed or reduced, things may bounce back. But if you are doing the same thing, let's say for the same, I think Upendra published a paper on bacterial diversity from CRRI in India, where he found some fertilizer application treatments over a 20-year period led to a different community. So because community composition, abundance, functionality depends upon habitat that they are residing in. If the fertilizer application is changing the physical chemical properties of the habitat, then it has a potential to change community composition. It is that actually that led to the suppression development by retaining stubble retention. Similarly with fertilizers, there's a lot of published evidence of fertilizer effects on denitrifying organisms, increasing the composition of certain groups and things, and linking that to denitrification. So fertilizer application like nitrogen fertilizer could change composition. It depends upon whether it is positive or negative, depending upon the magnitude of the effects and the frequency of the effect.
Another question, sir. You mentioned about the suppressive soils and also showed the importance of the involvement of fungi. Did you find similar significant influence of bacteria on suppressive soils?
Yes, maybe I missed one of the slides was bacterial one. I took a fungi as an example to describe in this presentation. But with bacteria, there was a difference in community, but not just community composition per se, but the responsive genera within the two communities to carbon addition from the roots or stubble is different between suppressive and non-suppressive soils.
Thank you. Thank you.
So in continuation to that, just by the addition of carbon, can you improve the suppressive nature of the soil?
Could you repeat the question again?
Just by the addition of carbon, can you improve the suppressiveness of the soil?
It's a very good question. I think I tried to make one point. not necessarily in all scenarios. In our case, the pathogens we were looking for, necrotrophic soil-borne pathogens. Necrotrophic pathogens depend upon energy availability, carbon inputs, biologically available carbon requirements. So for such things, that provision of these energy source carbon over longer periods of time through stubble retention, reduced tillage, led to the change of communities. But what we found is that that can be dictated by the presence of any physico-chemical constraints. And we found, other people have found, and tried to do the same work in calcareous soils of Eyre Peninsula, the ones in western part of South Australia. Annapurna, I remember we flew there. In those high pH alkaline soils, same carbon addition over 7 or 8 years didn't develop suppression. So we really couldn't understand why. But when we looked at this community composition and responsiveness, we found that in those soils, these putative suppressive genera did not respond to carbon addition due to the chemical and other constraints in these soils. So unless you remove those constraints, it seems that suppressiveness didn't develop. So not in all systems, addition of carbon alone would increase suppression.
Thank you.
Will it be depend on the quality of carbon or the nature of carbon molecules which are added?
That is also one of the hypotheses. And in those soils, they actually tried different types of carbon inputs. Cereals only, legumes and cereals and fertilizers, but it didn't matter which type of carbon you put in. So we also tried, one of my PhD students tried to transfer communities from a suppressive soil from eastern part of South Australia in pot trials to these calcareous soils and looked at, can we develop suppression? So although the community is suppressive communities, using 10% of soil microbes as inoculum, they didn't develop suppression when they were put into this chemically constrained soil. So it's not just carbon source, but the physico-chemical properties of the soil didn't allow the suppression to be expressed.
Okay, thank you. Thank you.
Thank you. In these suppressant soils, was there any abundance of fluorescent pseudomonads?
Pseudomonas flourisce in such a broad group. So they were one of the dominant proteobacteria, but the level of response in those soils to addition of carbon was very small. There was some, but very small compared to the other suppressive other soils from eastern part of South Australia and in Western Australia.
Thank you. Thank you.
I have a question in addition to this.
Yeah.
What foresee the success of soil transplantation experiments to increase the productivity of soils in agriculture system?
And the first part of it, I didn't get it. There was disturbance.
Sorry. I said, what will be the success of soil transplantation experiments in agricultural production systems?
It depends how much soil you want to transfer.
Not that. Very small amount to bring the soil microbial communities.
Which is what the PhD student tried. She tried to transfer 10% of the soil, because generally transfer of suppression experiments are done based on around 5% of the soil transfer. That's an established procedure by Ravira et al., Cook et al., and also we published a paper in 2014. So 5% is people consider as the demonstrating ability of transfer of suppression. I want to make a comment, Annapurna. Yeah. We seem to have quite a few students around different places. Are you getting any questions from them?
Yeah, there are.
Yeah. Some questions are from them only.
Kumar can now take over for the students' questions, please.
Yeah, it's all mixed only. We cannot discriminate who is student, who is faculty here.
Sure, that's
all
fine.
Interesting questions are coming. I guess they are from students only.
There's one question. How do you cultivate a healthy plant microbiome?
You have a healthy plant, it has a healthy plant microbiome. So,
there is another question from a student. who mentioned, who asked about Virome.
Student who is faculty here.
Sure, that's
All
right.
interesting questions are coming. I guess they are from students only.
Yeah, I student.
Cultivate a healthy plant microbiome. What is happening? It's getting repeated.
Plant, it has a healthy plant microbiome.
There's one question, Gupta. What about Virome from soil?
There is a very limited work on Virome, and in the last 2-3 years, there is a few papers, handful of papers, in terms of from the time of a type of sequencing required, and also interrogate the sequences, because when you are trying to annotate the sequences to a known member, whichever way, you do require a reference data sets and all. One of the limitations is the Virome-based data sets for especially soil-based Virome stuff are quite limited. So not, they're still, let's say, 10 years behind bacterial-based data interrogation.
Sir, I have a small question, sir.
Kumar,
In microbiome research, the bacterial exploration is dominating over other microorganisms. Is there any specific reason for that?
Oh, the environmental microbiome people all depend upon medical microbiomes. Most of the tools and techniques come from developments in non-soil based research. So, and in a way, bacterial taxonomy and databases are a lot more advanced than fungal taxonomy. A lot more people are interested in bacteria. So,
in connection to that, is there any specific marker for oomycetes already developed, established?
oh yes, there are at least 3 or 4 papers are oomycete specific primus. Yeah, you can search for oomycete specific primus, or yeah.
It is same like ITS or something else?
Oh, there are 2 types, but ITS1 and 18S1, both ITS and ATS based.
Okay.
Kumar, I have a question.
Gupta, there's one question from the student.
Yeah.
How to increase the beneficial microorganism in high pH soil and calcareous soils?
Oh, it is certainly an interesting, important question. Chemical constraint is the reason for lack of adequate abundance and desirable community composition, then remove the constraint. There are a number of ways you can alleviate chemical constraints, lime, gypsum, and variety of other ways. And whether you do the whole field soil or whether you perform these changes or make these changes in the root zone environment, that depends upon the level of change you want to see. But we should learn. That's one thing I should make a point to students and others. Just because you are doing new molecular tools and other microbial techniques doesn't mean that you should ignore wealth of knowledge and physics and chemistry associated with plant growth and agricultural fields. The new tools actually make it necessary to consider other aspects of the environment in order to be able to truly interpret the sequence data. It's very easy to make all these graphs with only the sequence data, but it means nothing. Your interrogation is very limited benefit if you are not able to relate why the differences are there, what is making those communities to respond. So it is necessary for young molecular biologists to learn basics about physics and chemistry.
True. So very true. There's one more question by Nisha Jaiswal. Shine 2 is basically used for Illumina. If we have done iron sequencing, then which tool we should go for?
either Andrew will answer now or Andrew will answer this after his presentation. That's
Okay.
more appropriate for Andrew too. Yeah.
Okay.
Can you hear me? I can have a go now.
Yeah, go ahead.
Yep. Chime 2 is fine. So it was developed around Illumina, I guess more specifically MySeq data, but it can be used for any of those data sets. I think on the documentation, there'll be some guidelines for dealing with ion torrent data and the error profiles and that. You need to be a bit careful with the data too, denoising as well, I guess, but there's documentation in those tools as well. So it should be fine.
Okay. Thank you, Andrew. There's
Madam, can I ask one question?
1 second. There's one question by Nisha Kumari Gupta.
Yeah.
Is metagenomics able to study functionality of all genes of non-culturable soil microbes?
Oh, if you know what all genes means, then you can say yes or no. But the data integration techniques are quite advanced now. I think this is another point that Andrew will certainly cover. And after his talk, you probably may not ask exact question. So there are limitations depending upon what you are looking at. If you go for a functional gene that is least studied and least known about it, then you probably may not be able to do as much. And then, like for example, nitrifying, NIF, AMOA genes are well-researched community. So from a metagenome data set, you're probably able to get a lot more out of that compared to a, let's just say, ACDS gene.
There's one question by Sajid. Is there any effect of different alkaloids on microbiome, mostly endophytes and alkaloid-rich plant like apple?
I couldn't give any first-hand information, but I have read papers in the literature of specific members of microbiome enriched in these plants of varying alkaloid composition, both medicinal plants and other types of plants. So microorganisms are ubiquitous, and also their capacity to survive in harsh environments is quite extensive. So there will be specific members of the community. Actually, the medicinal plant work mostly came from India, from I think Bangalore or somewhere there. And there's a specific members of bacterial biome that is more enriched in these medicinal plants with various compositional properties.
There's an interesting question by Mukhtar. How to apply microbiome concerns in hydroponic system?
It depends on how the hydroponic system is maintained and what stressors that are microbes in the systems are experiencing. So it's certainly an area of research that is gaining more attention, because in a number of countries, people are trying to grow plants indoors.
Yeah.
But you have to look at the principles that govern microbial survival, persistence, and capacity to function, and link that to the hydroponic system characteristics.
Both aeroponics and hydroponics are gaining importance now.
Yes.
One question by an anonymous student. I don't know why the student could not put their name. He says, or he or she says, I want to ask if any microbes are present in money plant because it increases very rapidly.
I'm not sure you guys can see, but you can't see it. I have a money plant growing that is 10 years old. It's only survived on distilled water, microbiology lab distilled water. For the first 10 years, it grew with no nutrients. But of course, as leaves die, new leaves come. And we actually looked at NIF-H community in that water. And we said, oh, it's getting all the nitrogen it requires. But as the leaves fall off and we didn't have any other nutrients, in the last 3 years, it is the root component is 80% of the plant. So money plant does have a microbiome inside, It does have a diazotrophs, but it requires phosphorus and other things.
Okay, there's one more question by Parmita. Is there any difference in diversity depending upon type of presence of macronutrients like N, P, K, C? Which one will result into highest species richness?
It depends upon which environment you are looking at. If you go in a, there's a lot of research published. I don't need to list any of that. A lot of it in the literature. If you go in those high pH calcareous soils where elements such as phosphorus and zinc are limiting because of chemistry, in those environments, general microbial activity responds to addition of P. But if you go in, higher organic matter soils from prairies in North America, USSR and northern India, alluvial soils, you may get only C with nitrogen, not necessarily with other nutrients. So the responses of different nutrients depend upon the habitat characteristics and the nutrient availability capacity.
One question by Anna Durai. How to identify the nodule microbiome?
I'm not sure what exactly they want. The technique to study microbiome of a plant part is to remove extraneous factors from that sample. If you are looking at nodule microbiome inside nodules, like many of you do, surface sterilize the nodules and do microbiome analysis. Similar to people isolate rhizobium, surface sterilize nodules and isolate rhizobium. Same tools. The microbiology principles required to study using microbiome tools are exactly the same.
Kumar, you have any more questions? I can see some more here. Another 5 minutes.
I have one question. Shall I ask?
Yeah, sure.
Yeah, Dr. Gupta, it's an interesting presentation. Excellent, especially the graphics. Nice. I have one question. How do you account for the microbiome contribution by the soil arthropods, one of the largest group of animals in the soils, especially like insects and their interplay and how they are modulating the soil microbiome?
Oh, the soil fauna microbiome interactions have gained quite a bit of attention during the last 3 to 5 years.
Yes.
And people have looked at microbes associated with individual microarthropod species and shown that they may differ depending upon which environment they are coming from. And much of this work is generally looking at look and see, not necessarily looking for any specific functional groups. There is a one functional group that is looked at for termites and other macrofauna, earthworms and things is actually diazotrophs, because people have seen or measured N fixation inside termite mounds and things. So there's quite a bit of research published from Africa, and I think a couple of examples from India, and a couple of examples from Queensland, University of Queensland, looking at diazotrophs and general communities inside different termite guts and termites. And also the microarthropod research comes from mostly from Germany and Austria, Eastern European work. So, but there is, no, at present it is just look and see who is there. Linking the functional implication of these community changes is a long way to go. It is a difficult task to interrogate because the effects of fauna on microbiome could be two reasons. Is because the macrofauna are changing the habitat, that's one way indirectly, or these fauna are selectively grazing or enriching through direct means. So to interrogate both of them and link to function is certainly a complex science, and number of new experimental approaches needs to be even developed to able to tease out the complex interactions.
Kumar, you have The more you can ask.
Yeah, I think we have
5
fairly
minutes.
covered most of the questions, ma'am. There are some minor questions are there. So about agriculture boom and agriculture productivity, I think not directly connected to this topic.
Yeah.
Otherwise, most of the questions are fairly answered and fairly discussed also. I think we can move on to the next session. That's my feeling.
All right. So thank you very much. Yeah, Gupta, you wanted to say something?
Maybe give people 5 minute, 2 minute time to get up and stretch before you start the next one.
Yeah, sure. But thanks once again. Excellent talk. And for all those interesting questions which got generated because of your talk, thanks very much. I guess we take 2 minutes break and then resume with Andrew's presentations.
Are we starting immediately now?
No, 2 minutes. 2 minutes
By
break.
12.
Okay, 12 o'clock we'll start.
Yeah, it's about
By 12 start.
Yeah, we'll start at 12 sharp.
Yeah.
Yep.
India time. I don't know what the time
India
is.
time, of course.
It's 5 minutes. So
Yeah, 5
Yeah,
minutes.
true. Start, ma'am.
Yeah, Kumar.
Can we start now?
See if Andrew is there or not. Is Andrew?
Yeah,
yeah.
I'm not seeing him actually in the list. Yeah, he has come. Yeah, yes, come.
Yep.
Can we start now, ma'am?
Yes, yes,
Sure.
please.
Okay, so it's 12. We'll start the second talk of this morning. The second talk is by Andrew Bissett. Those who are not too familiar with the name, Andrew Bissett is a very prominent scientist in Australian microbiome research, and he's going to speak on techniques for metagenome sequence data analysis. It's a very important topic. There were a couple of questions and a lot of discussion in the previous session, and about little bit about Andrews. Andrews Bissett is a senior research scientist with the CSIRO based in Hobart. And prior to this assignment as an important member of this Australian Microbiome Consortium, he was in Max Planck Institute for Marine Microbiology, and he is having a PhD degree from UTAS. And initially he joined in Canberra. Presently he is working in Hobart. So I request Andrew Bissett to deliver his talk on methods in metagenome analysis. Andrews, please.
Okay, great. I'll just turn my camera off if that's okay for the talk, because my bandwidth is a bit limited. I can turn it back on for the questions. So I'll just share my screen. Okay, so can everyone see that?
Yeah. Yeah, go ahead and.
Okay, great. So I'm going to talk today about techniques for metagenomic sequence data analysis. Really an introduction to metagenomics with a focus on amplicon sequencing. So the aims of today's session will be to introduce metagenomics and metagenomic workflows. And so to do that, I'll talk about experimental design, sequencing, and sequence analysis to a gene abundance table through annotation and a little bit about alignment. So we'll first start off with a bit of background. So what is metagenomics? It's a bit of a controversial question, actually, and I'm going to define it quite widely here, although I'm not trying to offend anyone by doing that. But so Wikipedia say it's the study of genetic material recovered directly from environmental samples, and the EBI define it as an umbrella term covering a range of studies based around high throughput sequencing, but they include whole genome shotgun sequencing, metatranscriptomics, as well as amplicon-based approaches. So a lot of people don't include amplicon-based approaches in metagenomic, the definition of metagenomics, but for today's purposes, we are. I'm also going to extend that definition a little bit further and include non-microbial DNA. So conceptually, what I'm talking about could be applied to any amplicon. So it could be applied to any eDNA study of larger things in the environment. I'm not going to talk specifically about those, but conceptually they're included in what we're talking about here. So why do we study metagenomics? In the context of soils in the microbial world, we mainly study them because a lot of the microbes that we deal with can't be cultured. A lot of them are cryptic in the environment, so we can't see them. They're hard to find. They're rare. And culture and capture-based investigations are expensive, so it costs a lot of money to go and do field surveys for fungi, for example. We can get a handle on those things using more high-throughput methods. Metagenomics allows insight into the ecology of microbes, so we can study them in their natural environment. So we can look at the way they behave and the way they interact, which we can't do if we isolate them. And the metagenomics tools allow us to analyze the natural structure of microbial communities. We can talk about some of the limitations of metagenomic studies. which I've misspelt there. So function can only be inferred from metagenomic studies. So it can be thought of as a potential function, even if we're studying transcripts in this case. If you want to know that the function is being carried out, you really need to measure that function. The viability and or activity of genes and organisms is only assumed. So we don't know whether the things are alive or not, or whether they're active or not when we're sampling nucleic acids. Numerical abundance of an organism doesn't mean that those organisms are the most important things for a particular process. For example, ammonia oxidizers aren't very abundant in soils, but they're pretty important in nitrogen cycle. So the spatial structure of the sample in the community is difficult to assess and is often ignored. So it's difficult to sample soils at the scale that a microbe would experience in the soil, so at the micrometer scale. And it's also difficult to know which organism a particular gene belongs to. We can have a guess, and some of the amplicon-based methods that we're going to talk about let us have a pretty good guess at that, but often we don't know, especially when it comes to genes that are on mobile elements or that are horizontally transferred. Gene assembly is difficult. I'd say not that possible in many of the sample types that most of you are thinking about today, soils, for example. And so we tend to base a lot of our analyses on non-assembly based methods, although Paul will talk a bit more about that. The numbers that we return from these studies are compositional, so they refer to relative abundance within a sample, which can be problematic when we're trying to infer whether something is dominant or not between samples. So different things affect the microbial community that is returned from any molecular survey or molecular study. The sampling design. So Gupta's already talked a little bit about that, but how many samples you take in a paddock, how many plants you sample, how many parts of the plant you sample are all important, and the system variability spatially and temporally needs to be taken into account when you're designing those experiments. The sampling method's important. So the size of the shovel you use, the size of the core you use, how you pull the plant out of the ground to sample the roots, the age of the plant, all of those things affect the inherent variability. The nucleic acid extraction method will have an influence on the type of organisms you encounter in your sequencing. One way to get around some of the variability associated with the extraction method is to use more than one method. Most people don't do this because it's kind of expensive, but it's one method that we can try. The sequencing method itself and the wet lab procedures, so the PCR, the specificity of the primers, the GC content of the organisms, all of these things affect the type of organisms that will be returned. And finally, the informatics workflow that you use, and we'll talk about that in a bit more detail. So there's a strong requirement to know your system and what type of data you need to address specific questions that you have. So today, I'm going to talk about a metagenomics workflow along the lines of this flow diagram here, and I've broken it up into different sections that kind of makes sense to talk about. So if we go through this flow, we'll start with experimental design, and then some kind of sampling and nucleic acid extraction. We can talk about those as all kind of upstream components of a metagenomics study workflow. And then we can talk about the sequencing itself, which is really a wet lab process, but we'll talk about it on its own, in its own right. And then the sequence bioinformatic workflow. So analyzing the sequences and producing some kind of data product that you will then use as input data for further statistical analyses and the workflow. And then other important considerations of any metagenomics study workflow are data storage and data sharing. Today, I'm only going to cover up to the end of the bioinformatic workflow component. It's kind of already quite a bit to get through in an hour. And we probably won't make it. So I'll go through each of these three components. So experimental design, sequencing, and the bioinformatic workflow. So there are several different types of metagenomic experiment, and broadly they can be broken into structural experiments or studies, or lines of inquiry, and functional lines of inquiry. And just repeated the image that Gupta had here from his talk, showing this concept of structure and functional analyses. And so structural analyses are really asking the question, what or who is there? And to address these kind of questions, we often use marker genes, so 16S, 18S, or ITS, or different housekeeping genes, or functional genes. So in a lot of the examples that Gupta was talking about, he spoke about the NHP gene, which is a gene associated with nitrogen fixation, and is used to infer the ability of the organisms that have that gene to fix nitrogen. We can also, so in the case of using a specific functional gene, so in the NHP example, we're targeting NHP, so we already know the function, but what we're interested in is the structure of the organisms doing that function. So which organisms are capable of that in our sample. In a functional type study, we're asking what might the microbiome be doing. So in a common example is whole metagenome shotgun analyses, or transcriptome, metatranscriptome analyses, where we're measuring the whole microbiome or the whole metagenome, and we're trying to annotate all the genes we find and find some indication of the important processes that we're finding in that sample. I will say, though, that there is a real need to measure the process to really address this. And I guess that's one factor that isn't quite in the infographic on the right here. So we have metabolomics and proteomics. So we're only representing the omics really in this, this process, but really to measure in situ function, we need to measure the process if we're going to infer that it's going on. And I guess if we include the inorganic metabolomic components, we could measure process rates. So to address these questions, you'll notice that largely we're talking about amplicon-based analyses. So, and by amplicon, I mean amplifying a gene or a region of interest, a genomic region of interest using PCR, and to produce an amplicon or a known fragment, or whole genome shotgun type analyses where we're just trying to sequence everything we find in a sample. So some of the benefits and non-benefits of those two methods can be seen in this table here. So we can go through this. So the cost of shotgun sequencing is much higher than amplicon sequencing, which generally leads to less replicates in a shotgun type study, and more ability to replicate heavily in an amplicon-based study. So amplicons in Australia run at about, I don't know, 50 or 60 Australian dollars a sample, and a shotgun sample would run at around $300 a sample. So there's quite a difference there. So in terms of sample prep, they're both similar in terms of the wet lab skills that are needed to do the library preps, but depending on the method used, less DNA is required for an amplicon sequencing study. Although if you do use some of the amplification-based shotgun methods, you can get by with small amounts of DNA as well. In terms of functional profiling, for amplicon studies, the tempting answer is no, but there are methods around that allow predicted functional potential based on phylogeny. And I think if you've got a well-defined system and you've got some shotgun data to back up what you find, and the genes that you're looking for are narrowly distributed through the phylogenetic tree, I think that that's quite a reasonable approach. But if you have genes that are throughout the whole phylogenetic tree, then it's more difficult. So trying to infer denitrification from 16S genes, for example, is pretty difficult, but trying to infer ammonia oxidation or methane oxidation is a reasonable thing to do, I think. In terms of shotgun analysis, yes, we can infer function, but again, it's only functional potential. So just because the gene's there doesn't mean that it's being used. For example, it's quite common to find NIFH genes in rhizobial species in Antarctica, and logically we could infer they're involved in symbiotic nitrogen fixation, except there's no trees in Antarctica, so clearly they can't be doing that. So you need to be a bit careful with those functional inferences, no matter which of these methods you use. In terms of taxonomic resolution, it really depends on the amplicon and the sequencing depth. So you need to make sure that that the amplicon you choose is informative across the region that you're sequencing for the things that you're interested in. It might be argued that amplicon sequencing is better suited to detect closely related ecotypes or variants because you can align all of the sequences you get back. So that could be one thing to consider. And potentially with the taxonomic resolution, because you're not restricted to a single gene, with shotgun sequencing, you may end up with higher resolution there. You certainly end up with more coverage generally because the libraries are a lot bigger. In terms of taxonomic coverage, it really depends on the amplicon. You can use primers that are universal, which will detect everything in your sample, or you can use primers that are designed to pick up a specific gene or a specific genera within within a certain gene. The shotgun sequencing picks up everything in the sample, but again, it's skewed to the most dominant things. And that's a pretty funny or a difficult thing to assess, really. So dominant doesn't necessarily mean numerically abundant. It means dominant in the amount of DNA you get per genome back. So if you've got a genome that's very, very big, you're more likely to hit it than if you've got lots of genomes that are very, very small. So genome size has a bit of an effect there as well. In terms of bioinformatic requirements, amplicon data analysis requires less. Shotgun analysis requires more. Not very well defined things, I guess. It depends what you want to do. And it's the same with the compute resources required. Generally, amplicon data sets are smaller and the search space in the databases is less. So you require less resources, more for shotgun analyses. Sensitivity to host DNA can be low for amplicons, but it really depends on the PCR primer of choice. The sensitivity is high for shotgun analyses, but can be overcome with either greater sequencing depth or depletion of unwanted reads. And in terms of bias, I guess it's commonly accepted that the bias is medium to high in amplicon sequencing. I mean, that's why we do it. We're biasing our analyses to retrieve the specific data we're interested in. The shotgun data, there's lower bias, but inherent in that is a lower resolution towards a target if we have a target that we're trying to sequence. So I'll talk briefly about experimental design. And I think that this is something that's overlooked or not people don't spend enough time on this aspect of metagenomic experiments. But the research design is the foundation of good research, and it really does direct research planning and collection of data. It defines a statistical analysis of that data, and it guides the interpretation of any analysis results. It should be the first thing that you do always. As a side note, it should also be the first thing that's articulated in the methods section. So the experimental design should be explicitly stated there. It's kind of infuriating to read papers that don't do this. And when it's done, it's really difficult for readers to interpret results and analyses. So often in a methods section, the emphasis is on statistical methods. And while they're important and need to be described, they're only one component of the design, but it's really necessary to know how and why data was collected in order to assess any of the applied statistical methods. So common steps in study design are laid out here. First, the question is defined, and then some hypotheses are defined. And then it's good to describe what you'll measure and observe, and then to reflect on whether those things that you're going to measure and observe will allow you to answer the question. And then once you get there, you can design the study, and it's good to consider data lifespan. So we'll just go through each of those things. Defining a research question requires moving from the general to the specific. So it requires going from general ideas or concepts to defining a single question with a potential yes or no answer. So further clarification of the intent of the research may be made by reframing the question as a specific objective in that step. This process is often difficult, and moving from the general to the specific is difficult, but it's always worthwhile. So after we have the question, we move to defining the hypotheses. And so the hypotheses will come directly from the question defined. And the hypothesis is simply a clear statement of the expected outcome of the experiment. So it'll be based on whatever evidence is available. So the more novel the question, the more limited the evidence. And so your hypothesis might be based on nothing more than a hunch. But if the question is based or comes from years of research, you might have a really well-informed hypothesis. And so the hypothesis will allow the researcher to articulate exactly what they'll need to measure in order to test it. So after we have the hypotheses, we can describe what will be measured. So what data will be collected. And then after you work out what data to collect, you'll be able to answer the question, will it allow me to answer the question, to answer to your question. And so in terms of a metagenomic type study, here are some ideas of, ideas to consider when addressing the what data will I collect question. So it's important to know what information you want from the amplicon. So this includes to know whether the gene that you're sequencing will allow you to differentiate the organisms that you're looking for, whether the region you're sequencing will allow you to put the organisms in a phylogenetic framework, so into a tree. You also need to know what other information you need from your sample. So if you're interested in looking at nitrogen cycling or denitrification, for example, then you might need to measure nitrogen. You might need to measure the denitrification rate. You might need to measure the soil pH. So all of these things that you need that aren't sequence data to let you address your question. You need to know how much material you need to get to do all these things. So if you're extracting DNA from 10 grams of soil, then you need at least 10 grams. If you're then trying to assess carbon, nitrogen, phosphorus, sulfur, pH, particle size on the same material, then you need more than 10 grams. So you need to make sure that you've got enough material, that you're going to collect enough material. Can you afford to have any missing data? So is the statistical analysis you're doing robust to an unbalanced data set? If you can't afford to have any missing data, then it's better to collect more replicates than you need, because it's pretty common to have missing data in data sets. So what format will the final data be in, and how will you use it? Do you have the compute capacity to use that data format? Does the plan that you have conform to minimum information requirements? So is the information that you're going to collect enough to allow you to make the data publicly available and to be reused? And is the data that you're going to collect with the sample enough to publish the data? So there are minimum information requirements to publish data on NCBI, for example. So you need to make sure you collect those or you won't be able to publish your data. So are false positives or false negatives a concern? How much coverage do you need? So if you're interested in a specific gene in the environment, how deep within the environment do you need to sequence to find that gene? What controls do you need? What experimental controls and technical and analytical controls do you need? And how will you use the controls to inform decisions? And then finally, is there anything else that you could easily collect that would make the data that you have more generally useful? So, you know, if you're going out and you can collect an extra piece of data cheaply, so you might be able to estimate plant cover, for example. You know, you just write down a percentage on a piece of paper and then submit that with the data. That's data that would be useful for someone else to reuse the data, and it's easy to collect. So it's a good idea to collect that data if you can. So after you've answered the first three questions, you can go on to design the study. And so the study design will be driven directly by the hypothesis and what you'll measure. At this stage, you should be trying to reduce sources of variation and identify known and expected sources of variability in the experiment. And finally, it's good to think about data lifespan. So trying to make data fair and to make the data reusable and your results repeatable. So I just thought that we'd go through an example looking at variability in the data you need to address a question. And this seems like a trivial example, but as you'll see, it's not quite as trivial as it first appears. So the question that we'll be trying to address is what is in sample A? So what organisms are in sample A and what is their abundance or relative abundance? So we can start by describing the system and the data that we'll need. So in this case, the sample we have is a commercially available mock community. So it's from the American Type Culture Collection. It's MSA 1002, which is a 20 strain even mix of organisms. So what do we know about the system? We know it's bacterial. We know that there are 20 organisms in it, and they're listed here. These organisms are all different at the greater than species level. So most of them are actually different genera, but there's a couple of Staphylococcus and Streptococcus that that are different at the species level and not at the genus level. We know that that in this mix, it's whole organism DNA. In this case, our system's well characterized. The genomes are available, so we can download them. We also get documentation with this this mock community telling us what's there, and a validation, like a report telling us that that they've tested it and what they say is true. So we're also expecting low variability at the sampling stage. So because this is a commercially produced, then it's highly controlled. We're expecting every vial of this to be the same. And then so any variation we see in our sequencing, we're expecting to come from the sequencing process or the wet lab library prep lead up process rather than from the manufacturer in this case, or the sampling process, I'm calling it. We also know that the community is very even. So you can see here that this mock community consists of 5% of all of these organisms. So an interesting question to ask and think about, I guess, is what does 5% mean? It's quite difficult to find out from the documentation here. So it could be 5% in terms of organism counts. It could be 5% in terms of DNA. We're not 100% sure. So we'll assume that it's 5% in terms of well, let's assume it's DNA content. So what do we need to answer our question of who is there? We need a way to classify and count the bacteria. So we could do a whole shotgun survey, but it seems like it's not the best way to do it, or not the most resource efficient way to do it. we could choose a gene that we know is in all of these organisms and sequence it and count it. So 16S rRNA gene is commonly used for this kind of activity. So we'll use that. We could use another gene, but for the purposes of this exercise, we'll use 16S. We need the gene to be able to differentiate the species from the chosen fragment. So we need to choose the region of 16S that allows us to differentiate all of those organisms. Because they're not that closely related, in this case, it probably doesn't matter which region we choose as long as we choose a variable region. And so we're going to sequence the start of the 16S gene. We also need a database to compare the gene, the sequences to. In this case, the genomes are available, so we can construct that database ourselves. And then the second part of the question is, what is the relative abundance of these organisms? So we need a way to count the organisms. And our first assumption is going to be that we're just going to produce a gene abundance table. So we're going to look at the number of reads that hit each of those 20 things, and we're going to count them. So that's a reasonable first assumption. So this is real data that we have through the Australian Microbiome Project. We've sequenced with the Illumina MySeq 27 forward to 519 region of 16S gene with 300 base pair paired end reads. We've gone through QC, denoise and annotation process. It's a pretty standard workflow. I'll go into a bit more detail about that later. We've done 15 sequencing runs, all from the same batch of the mock community. And each run had various other samples on the sequencing run. So around 100 other samples. It actually ranged from about 94 to 123, I think. So if we just take the gene abundance table, like we first, or like I first suggested, we get a different number of reads per sample. So this bar graph represents, each bar represents one of the 15 sequencing runs. You can see the different expected organisms here. So there's 20 of them. And you can see the number of reads returned. And so if we were just to count the number of reads in each sequencing run, we would think that sequencing run J654K has a lot more of our bacteria in it than J6H7B. Now we know this isn't true because they're exactly the same community. So in this case, we can demonstrate quite clearly that the number of reads that come off the sequencer per sample isn't such a great indicator of how many organisms were in the original sample. So what does influence the number of reads in an individual sample off the sequencer? Essentially, it's the number of samples on the sequencing run divided by the number of reads that come off the sequencer. So if the sequencer produces 100 reads and you have 100 samples, you would expect 1 per sample. If there are 10 samples and there are 100 reads coming off a sequencer, you would expect 10 per sample. This assumes that whoever was doing the library prep was able to mix the libraries in equimolar amounts. And we know this is really difficult. And so what we see is the average reads per sample is always the number of reads off the sequencer divided by the number of samples, which equals 20 million divided by 100, say. And that never changes, but the variability around that does. And generally the variability around that is really, really high. So it's, it's X plus like 1000X minus 50X or something. So it's like the variability is really high. So how do we get around that? Well, one way to get around that is to look at the relative abundance. In this case, the bars again represent the sequencing run, but instead of the number of reads on the vertical axis, we've got the percentage of reads in that sample. And we can see I've only represented the 20 target organisms in the bars. So none of the white space at the top of each represents the unmatched reads. So in this sample here, around 70% hit the target, and there was around 30% of things that shouldn't have been there. In this sample here, we see that it's much more as we'd like. So very few unexpected reads. And so again, we can see that the relative abundance in the total sequencing run hasn't really told us much about the relative abundance of those organisms in that sample. It has given us an indication of contamination or crosstalk between those samples, but it hasn't told us much about the number of reads. So how do we make that better? Well, we can just look at the target sequences. So in this case, we're only interested in the organisms that should be in the mock community. So we can see that all of our sequencing runs now sum to 100%, and we can see the relative abundance of each of the 20 expected organisms. What do we notice about this? We notice that some of the organisms are more abundant. So this Clostridium here. And we know that some of the sequences are less abundant. So this orange one here, or this pink one up here, where they should all be equal at 20%. So why could this be? Well, in this case, we've sequenced the 16S gene. Now we know that the number of copies that each organism has of that gene isn't one. And so some organisms have lots of copies of 16S and some organisms have less copies of 16S. And so we're trying to make this on a per organism basis. So we're trying to count it on a biomass basis, I guess. So we could try and correct for the copy number. Should be simple enough because the ATCC give us the copy number in their documentation, one would expect. Although when you look at the documentation for this well-characterized mock community, it's actually quite hard to find the copy number. So the ATCC have a poster which describes the this mock community, and it reports the copy number per organism. And you can see I've put those in here. So the most is in this Clostridium. The least is in the Helicobacter and this Actinomyces. But if we download the genomes from ATCC and pull out all the 16S genes and count them, we can see that the copy number they report is different to the copy number held in their genome repository, which is kind of interesting, although a little frustrating. Some of the genomes are actually annotated. So instead of going to the sequences and pulling out the number of 16S's, instead of going to the genome, the assembled genomes and pulling out 16S reads, there's some metadata annotation telling us the copy number. And interestingly enough, the copy number in the annotation matches the copies I found in the genomes, but not the copies in the posters. You'll see that not all of them are annotated. So even finding the copy number in this system is somewhat difficult, which was actually quite surprising. But if we correct for the copy number, we can see here that this is the image from the last slide at the bottom, and we've got a new image at the top here. And so this is the copy number corrected data, which we're hoping will become closer to 5%. And you can see it's changed the numbers a little bit. So it's compressed some of these bars, these bigger bars, expanded some of the smaller bars. And it's a bit closer to an even distribution actually than previous, but it still doesn't reflect the 5%. Why not? What extra information do we need? And so you can see how difficult it is to conduct an experiment when you don't understand the system and when you don't know up front what information you need. And even in a really simple case like this that should be quite easy to deal with, it's quite difficult. The extra information that could help us here is the genome size. So some of these genomes are quite small and some of them are quite large. And so the amount of DNA going into each, the number of organisms going into the mix when you're mixing an equal amount of DNA is different depending on the genome size. So we haven't taken that into account in our analyses. And also there seems to be some kind of bias against this bacillus here. So this was a high copy number organism, and yet we don't see it very much. So we probably want to go back to our system and make sure that our primers were actually not biasing against us picking up this bacillus. And so I'll leave that example there, even though we didn't quite get to the end of it. But it was just a demonstration of how important it is to try and understand the system before you start. So study design is the first thing you need to do, and it's probably the most important thing you will do. And it does guide all downstream aspects of the study. It's not a linear process, so you'll often go back and try and collect more data or try and do things a little bit differently. But you should know what you need to address your question, and you should know what data you'll need to utilize the statistical methods you'll use. Ideally, you would do some kind of pilot study. And I think that these days it's actually quite easy to do that, because it's pretty rare that you would be sequencing such a novel environment that there wouldn't be some publicly available data from a similar environment for you to look at first. So we'll move on to talking a little bit about some sequencing. So I'll cover briefly throughput, or the number of reads, the read length, and some controls. So because Illumina is the most commonly used platform at the moment, that's what I'll be talking about. The concepts around fragment length and read length controls apply to other methods, but I'll be using Illumina as my example. So in terms of throughput or depth, amplicon versus shotgun, it really depends on the question you have and whether or not your gene's likely to be high enough abundance to be present in a well-represented way in a shotgun library. Gupta spoke about that a little bit, and I'll talk about that a bit more. If it's not going to be high enough in abundance, you would want to consider amplification. A coverage curve could help you assess the adequacy of the depth you have. And the other question that that you need to address in the amplicon versus shotgun world is, do you need to align the gene region to differentiate with most environments? So how do you centralize genes and enough coverage? I could also consider enrichment of the community to try and bias towards picking up the genes you're interested in. And as I said before, a pilot study is a really good idea, and probably even easier now, as I mentioned, is data from Sequence Read Archive, for example, from a similar environment. So the basic rule of thumb is that for read depth, more is better in terms of coverage, but in terms of compute and sequencing costs, more is not better. This is similar to the example that Gupta showed earlier. So we have here four different samples from different environments, different regions of the world, and all of these have been shotgun sequenced to varying degrees. I've got the total number of reads. So around 14 million, 17 million, 6 million, and 11 million reads for each of those environments. The number of assigned reads is also listed there. And you can see that typically around 35–40% of the reads were actually assigned to known genes. When we look at the types of genes, you can see that some are reasonably well represented. So as Gupta mentioned, around the carbon cycle, probably in the phosphorus cycle, and these housekeeping genes are well represented. But if you're interested in ammonia oxidation, for example, then these shotgun libraries aren't much help with 3 reads in the rice paddy, for example. It's the same for NIF-H. The numbers are really low. So if your idea is to study nitrogen fixation in these environments, then shotgun libraries aren't probably of that much utility in these environments. So having an idea of what genes you're likely to find and in what abundance in your environment would be pretty useful. And you can see that all of the patterns here are kind of the same. So these genes are in low abundance in all of these samples, even though they're from wildly different areas. So a wheat farm or a wheat field, actively growing wheat field, a dry desert soil, a wet, probably anoxic rice paddy, and a grassland. And so, so you can see that you don't have to go too far to get some pilot data to give you an idea of what you'll need to do. So I'm not going to talk too much about read length. I think Paul will cover that in his assembly type talks. But there's a difference between read length and fragment length. And so the read length is dictated by the sequencing technology. So typically on Illumina MySeq, it's out to about 300 base pairs per end. The fragment length that you can sequence is, if you want to merge those reads into a single amplicon, essentially around double that length. If you're looking at assembly, you can sequence from paired ends from quite long fragments and quite short fragments and try and put them together. So trying to work out some balance between the coverage and the read length and your ability to merge reads in an amplicon sense or assemble reads will dictate read length versus fragment length. For an amplicon, a longer fragment equals more information, although a longer fragment equals less merge overlap. So if you have a 600 base pair fragment, then in 300 base pair, to paired end 300 base pair reads, your merge to put those reads together is 0. You're essentially concatenating them. If you have a 300 base pair fragment and you do 300 base pair sequencing, then you're going to end up with two times coverage across that fragment. And so if trying to call the base pairs at high confidence is important to you, then that 2 times coverage could be really important. So just to recap on where we've got to, so I can remind myself, we've talked a little bit about experimental design and a little bit about the sequencing process, although not too much there. And I'll just spend the remainder of the time going through bioinformatic workflow. Again, I'm not going to touch on these downstream areas here, but I guess we can answer some questions about them if people have them. So in terms of informatic workflow and sequence analysis, there are many automated workflows for both amplicon and shotgun type data. I've listed a bunch of them there. So Galaxy, Chime2 has been mentioned already. There's the NF Core AmpliSeq pipeline for amplicon data. Squeeze Meta and MetaPipe deal with shotgun type data. Magnify through EBI and the MGRAS platforms deal with both amplicon or shotgun data, and there are many more. So most of the times these work, but you need to be aware of that they will provide output no matter what you put in, and so the output may or may not be sensible. So understanding what's going on is a useful thing. So I've taken this picture from the Encyclopedia of Metagenomics, funnily enough, and it shows a typical workflow for next-gen sequencing data for a marker gene analysis, so an amplicon-based analysis, or a shotgun metagenomic analysis. And you'll see that these two workflows have similar conceptual steps. So essentially there's some QC, there's some assembly and denoising, and then there's some annotation and statistical analyses. So interestingly enough, in terms of dereplication and contamination removal, they're only listed in the shotgun section here, but really they're applicable to the amplicon data. Dereplication is probably even more applicable to the marker gene analyses, where you end up with a lot of reads that are identical. In a shotgun study of soils, for example, you would expect very few duplicate reads. If you're getting a lot of duplicate reads, then there's something wrong with your library generally. Contamination removal is pretty important in marker gene analyses as well, especially under the Illumina platform, where it's spiking with things like PHI-X to improve diversity across the plate, across the sequencing face. There's a step listed here in the metagenomic analyses around pulling out ribosomal RNAs and doing taxonomic calls against those, and also calling genes. I've kind of put this upstream of the, the arrow should really go upstream of the data generation. So in the shotgun example, pulling these genes out is done bioinformatically. In the marker gene survey, effectively we're doing that enzymatically. So instead of using the database or HMM profiles to pull out the ribosomal genes, we're using PCR primers to pull them out. So we're doing the same step, we're just doing it enzymatically. The statistical analyses and visualization of results for some reason isn't listed in the shotgun metagenomic side, but I think that most people generally would do that with their shotgun data. And finally, one thing that's omitted there on the shotgun side of things is that especially in quite diverse environments like soils, there's not always an assembly step. Often we're just taking the reads and mapping them against functional gene databases to look at what genes are there in a gene-centric type analyses. There's a bit of a NETA diagram. So this is from the MetaPipe pipeline, which is supported by the European Elixir community. And again, it's the same kind of thing. We take reads, we may or may not do an assembly. We can do taxonomy classification against the 16 S's that we pull out. If we do assembly, we can try and bin them and produce whole genomes or metagenome assembled genomes in this case. Otherwise, we can take our contigs or our raw reads and put them through a functional annotation tool and produce annotated genes. So Paul will talk more about these tomorrow. So if anything, the take-home message I want people to have at the end of this is that you should have a clear idea of what you want to do and why, and you should be able to check the input and output of all of the steps that you're running, and you should be able to make sure that it's sensible. You should be able to explain, or at least understand conceptually, how whatever algorithm you're running works. So at least in a broad sense, even though you might not understand the math behind it. And if you can't do those things, then I'd strongly suggest that you need to engage someone who can do those things for this part of the work. It's pretty easy to put data into a computational algorithm and have data come out. But often that data can be nonsensical if you don't know how to check it. So in our amplicon example, the amplicon workflow is the simplest and easiest to step through. So I'll concentrate on that, and Paul will kind of expand to the shotgun type analyses tomorrow. And so conceptually, the amplicon analysis will comprise getting your sequences, doing some kind of quality control on the sequences, removing repeated reads, denoising, mapping your original reads to the denoise reads, annotating the sequences. So we can annotate without doing any of this as well. So annotation can just be on the raw sequences, or it can be on the denoise sequences. And it does require a database, and it can be done via a bunch of different methods. Finally, there's often some downstream QC, and we're going to produce an abundance table. So the way I'm going to step through this process is, again, to address this question of what is there and how many are there. And this is the question that we used with the mock community example previously. And so the aim is to produce this organism abundance table, and then from that organism abundance table to produce the relative abundance type images that we looked at earlier. So we're going to start with sequences, in this case, PCR amplified sequences, and we're going to end up with a table like this, where the samples are the column headers and the organisms are the row variables, and there's a number in each of these cells. So the number is often 0 in the case of microbiome data, but there's a number here. This table could be transposed and be the same. If, for example, you like working in R, then you would usually have this table transposed so that the organisms are up here and the samples are here, but it's the same. So as soon as we design the experiment and we know how many samples we're going to take, we can fill in the column labels of our table immediately after experimental design. After classification or annotation, and including denoising in this step here, after we've denoised our data, we can fill in the row labels because we know what sequences or what organisms are there. The final step then is to build the abundance numbers, and we do that by mapping the original data back against these row labels or the sequences that are represented by these row labels. And so I'll go through each of these processes, but essentially we're trying to build this table, and we can fill in different parts of the table after different parts of the process. So broadly speaking, QC or quality control involves following steps. So make sure you're happy with the sequences quality generally, and that the quality of the sequences will allow you to do what you need to do. So you want to remove the unwanted bits, so potentially low quality bases, adapters or primers, and non-target sequences, for example, host sequences. And you want to merge paired end reads that will merge. I've kind of put that into the QC step there. So the process isn't necessarily linear, and you can do different parts in different orders. So for example, you can QC and the low quality bases and denoise before or after merging paired end reads. And you can remove host reads first or at the end. So if you don't have very many host reads, you can leave it till the end. If 90% of your data is host reads, then you want to get rid of them up front so that you don't have to spend so much compute resource on dealing with them throughout the whole process. So what I find it's good to do is to draw a picture of how your data is going to look, or how you think it's going to look. And then you can kind of step through the process iteratively. So we start here with a DNA extraction. We end up with a whole bunch of genomic DNA. We do a PCR and we amplify the 16S gene. And so each color represents a different organism here. They're all the same length. They're all roughly the same length because we've amplified a specific fragment of 16S. We sequence those with MiSeq, in this case, MiSeq 300 base pair paired end reads. And so we end up with a read 1 with some junk on the end and a read 2 with some junk on the end. We merge those together, we do some quality control, and we end up with our fragment. And so this fragment here represents all of these fragments here. So we're not just ending up with one, but we're ending up with a bunch of them. So in a bit more detail, we start off with a long piece of DNA. We have our region of interest specific primer. We anneal that to this long piece of DNA. We do some PCR and we amplify this bit in the middle. We then attach some indexes and some Illumina chemistry on the end, these P5 and P7 adapters, which allow the fragment to stick to the sequencing plate. We then normalize and pool the libraries, so we can put a whole bunch of them together and pull them apart again, and then we sequence them. And so what can that look like in the end? So this, these three diagrams here represent the three possibilities that we have in terms of merging our paired end reads. So the insert size is represented by this arrow at the top of each of the three. And if our insert size is greater than the length of the number of cycles, so in this case we're doing a 300 cycle run, so we end up with 300 base pairs from this end of the read for read 1 and 300 base pairs from this end of the read for read 2, and we produce these two blue bits. And you can see that if we have a fragment, an insert size or a fragment length greater than 600 base pairs, then we don't end up with any overlap. And so we can never merge these reads into the single contiguous fragment we want. There are times when you don't necessarily want to read to merge the reads. For example, if you're sequencing ITS1F to ITS4, so ITS1 through through the 5.8 region and ITS2, the 5.8 region is highly conserved. And so you might only want this bit, and you might not want this bit. So this is a valid thing to do. But you need to be aware that there's no point trying to merge them. Your merging step will fail. The next case to consider is that the insert size is less than the length of the number of cycles, and that there is an overlap here. And so you can see here that we've sequenced from this end with read 1 and this end with read 2, and the two reads overlap, and so we can align them together and produce a single read that we want. And so this is the most common scenario that people are targeting with amplicon sequencing. The third example, which is also kind of common, people don't mean to do it generally, but it's kind of common if you're using shorter insert or fragment lengths. So for example, 18S V9 region is kind of short, and if you sequence it with 250 base pair paired end, you'll sequence off the end. So you can see here that the read 1 sequence goes through the target region and into the fragment specific primer, and then through the barcode, and then through the adapter, and the read 2 does as well. And so when you try and put those together, the fragment that you're assembling or aligning is going to begin with read 2. Now, a lot of aligners don't like that, and so you need to make sure the aligner or the merger that you're using is happy with that configuration, or it'll tell you that it couldn't merge any of your reads, and that'll be disappointing, I guess. So when we look at sequence quality, typically this is the quality score for the sequences is represented with these numbers here. These are the bases along the 300 base pair read, and you can see that the quality drops off as we go along the read. It becomes less, and this is even more pronounced in the reverse reads. So if you're trying to get reads to merge and align, you need to make sure that you have enough overlap to get through this poor quality region that you might want to trim. So just as an example here of the difference it can make trimming reads before and after you merge. So those of you who use data to do the denoising, for example, will know that the denoising and the QC is done before merging rather than after. And so in the upper example here, the green sequences are good quality, and the reds, the green bases are good quality, and the red bases are poor quality. However, we've defined that. And you can see because the fragment overlaps completely, even if we were to trim the red bits off, we would still have a reasonable overlap here, and we would be able to produce the consensus sequence, which would be this bit. So in this case, it doesn't make any difference whether we trim before or after merging, or we control quality or denoise before or after merging. In the lower example, though, you can see that if we were to trim all of the red reads here and here, we wouldn't end up with any overlapping region. Now, you could argue that because these reads are poor quality, we don't want to use them, but the quality of the individual read of this individual A, for example, is less than our threshold. But in this case, we have 2 times coverage. So we actually have 2 A's. So if this, say if our quality cutoff was 20, if this was 18 and this was 18, since they're consensus, our consensus quality is actually greater than 20. So doing the trimming after you merge is a better option in this particular case. So you need to be aware of what's happening when you're doing these steps. So one important question to ask is, do you need the quality scores after you merge? I would say you probably don't, but different tools deal with the post-merge quality scores in different ways. So some of them report the largest score of the merge pair. That's the most common, actually. Some report a sentry value to notify a mismatch, and this value is commonly a 2. So if you see a 2 in your quality score after merging, that's not the real quality. That's basically the algorithm telling you that there was a mismatch there. Some of them remove the quality scores altogether, and some of them calculate a new score, a correct quality score. And I'll just quickly show you the implications of that. So this figure comes from Robert Edgar's USearch website here. So we have 2 sequences here, and we have quality scores here below for each base pair, a forward read and a reverse read, and these are the quality scores of each of those reads. And you can see here that we have a C and an A, and there's no overlap there. So we're happy with the 32 and the 34 there because we don't have any more information. This T and this T are high and low quality, but essentially it's higher than 20 because there's another one of them. So some algorithms will report 20, some will report 2. The USearch option here will report the newly calculated quality. And you can see the same in the mismatch here. So some algorithms would report a 20 in this case. Some of them would report the sentry value of 2, and some of them will report neither, but the actual quality that should be reported is 16 in this case. So it's less, because there's a mismatch, it's less than 20 for the T, but the T is preserved because it is the highest quality score in this case. So it's important to know what's going on with those things when you're merging reads. So the final part about QC. So if you want to remove adapters or primers, you should be removing adapters or primers, and you can do that by specific sequence searches, or you can do a hard trim. So if you know the primer is 18 base pairs long, you can just cut 18 base pairs off the end of the sequence. You can also remove sequences that are too long or too short. For example, if you know your fragment length should be around 500 base pairs, if you get a whole bunch of stuff that's 200 base pairs back, you can check it out and see how likely it is to be real. And if not, you can remove everything less than a number of base pairs. It's common to remove sequences that are returned with any ambiguities or ends as poor quality. And it's common to remove sequences with long homopolymer runs. Although with Illumina data, the long homopolymer run is likely to be an N, which will be removed in the ambiguity step. The homopolymer run removal is kind of a hangover from 454 sequencing, where that was a common error. And it's common to try and remove non-target or contamination, or non-target really. So PHI-X or host, for example. After you've done this step, you'll have a bunch of sequences that you're happy to proceed with. And then we can go ahead and try and fill the row labels in our abundance matrix. We do this typically by denoising the sequences. So denoising is really a computational method for removing sequence errors from the amplicon reads in an attempt to identify real biological sequences. What happens in the PCR step is you introduce a whole bunch of error, and in the sequencing process, there's also a whole bunch of error. So quality score of 20 is about 1% error rate. So in 500 base pair read, you would expect around 5 errors just from sequencing. And so the way this is done is conceptually at least, we take the most abundant sequence and we take sequences near that sequence and see whether their relative abundance or their abundance relative to the initial sequence is indicative of this sequence being real or being one of these. And so what we try and do here in this image, the real sequence is represented by the larger circle, and the errors around that are represented by the smaller circle. And so we're trying to error correct these ones into this one. This image is taken from the Data2 paper, and it's not the image isn't quite correct, because what happens in reality is that you don't end up with just this one red circle here. You end up with the one red circle and a couple of these other ones anyway. So it's not a perfect process. And it really is a balance between splitting and lumping the data. So it's essentially some probabilistic estimate of the real biological sequences. And as I just mentioned, all of the methods are some balance between lumping and splitting. The appropriateness of the algorithms depends on the sample and the community under investigation. So there's no universally right way to do it. If you're looking for a specific sequence in the data, you don't need to denoise. So if you're looking for a specific sequence or type variant in your data, in fact, if you do denoise, you might lose that variant. If it's denoised, if it's rare, it could be denoised and become something different. So if you're after a specific sequence, you're better off to search the data set without denoising. The other question to address here is whether to denoise on a per sample or a per sequencing run basis. So per sample is potentially faster. You can run all of your samples in parallel, for example, if you have enough compute cores. There's potentially fewer crosstalk issues, although you also miss quite a lot of the rare biosphere by doing it. You only need the data from one sample. So if, for example, you're pooling sequencing with some of your friends at the local sequencing facility and you only get your data back, then it's not such a problem. If you only get your data back on the sequencing run, then analyzing it per run is not an option. You will get fewer ASVs total per run, although you may not get fewer per sample. Doing it per sequencing run is better for detection of rare organisms and chimeras, but you do need the data from the whole run and you do need a little more compute. Grant. So I will say that most of the algorithms recommend some approximation of per run denoising. So Data2 by default is per sample, but it's based on computational limitations rather than any other real reason. They do recommend you use their pseudo pooling method, but yeah, really per run is likely to be the way to go. So conceptually, an ASV or a ZO2 is a cluster without a defined radius. So it's not that different to producing a hard radius similarity OTU. It is a balance between lumping and splitting, as is defined radius OTU clustering. The entities or the sequences that comprise the rows of the matrix should be as good a quality as you can get. So generally, we only allow things that are represented above a certain threshold to become ASVs. So singletons in the whole data set can't become an ASV, for example. And the ASV is the highest resolution data we'll get, although it is clustered. So we could use the unique sequences, but computationally that's difficult to deal with. And from ASV data, it's also possible to produce lower resolution data, so defined radius OTUs or taxonomy, and then switch between any of those resolutions. So all of the denoisers work similarly. I'm not going to talk about or recommend a particular one. Which one you choose really likely depends on your compute resources and the requirements for QC before and after merging, and really what you think about per sample or per run data. But they're all of the modern or current denoisers are really quite good. So we've designed our experiment and we've filled in the column labels. Now we've denoised our data, and so we've started with our full data set, and now we've got a list of what we think are biologically real sequences. Now we want to fill in the numbers in our table. So we do a step I'm calling mapping. And to do that, we map the original reads to the identified amplicon sequence variants or ZO2s, and that allows us to fill in the numbers. So how does that work? We essentially take all of our original sequences and we look for them in some database. In this case, our database is our list of known organisms. And if they exist, we call our sequence that thing. So we take our first sequence and we say, is it organism 1, 2, or 3? If it's 1, 2, or 3, if it's 1, we put a number here. We take the next sequence. If it's 1, again, we put a number here, and we sum those numbers at the end of the process. The important things to consider here are how the search is going to be conducted, local versus global alignment, and the search order, and how the search will be terminated. I won't go too much into local and global alignments, but if you are doing database searches, it's important to note that some search algorithms will align across the whole sequence space, so including terminal gaps here. And so in the global alignment of our sequence here, with matches indicated by these lines, we have 7 out of 14 possibilities matched. So our similarity using a global method would be 0.5. We have this semi-global method where we're considering everything except the terminal gaps, kind of. So in this case, it would be 7 out of 12. So it's 0.58. And in this case, we only consider the locally aligned region. So the region from the first aligned bit to the last aligned bit and ignoring terminal gaps. In this case, we get 7 out of a possible 10 comparisons, so 0.7. So you can see that 3 different methods aligning the same sequences give us sequence similarities between 0.5 and 0.7, so 50% and 70%. And so considering which alignment method is appropriate for your question is important. And the other thing to consider is the input order of the search space, and the termination conditions. So are you searching the database randomly, or are you optimizing the search order to search the most likely candidate first? And if you're optimizing the search order to find the most likely hit first, then you can terminate the search much more rapidly if it finds a hit or if it doesn't find a hit. If you're putting the sequences in a random order, you kind of need to go through the whole database, which takes a lot longer if the search space is large. So you're essentially asking your algorithm to find your sequence among a whole bunch of sequences, and you're giving it some termination conditions. So you're telling the algorithm when to say it's found something that matches and when to stop searching because it's been successful or unsuccessful. So if we're searching at 97%, if the sequence similarity is greater than, I've put that the wrong way around, it's greater than 97%, then we call it a hit. And if it's less than 97%, we call it a miss. So if there's a hit or a miss at the first comparison, so the first sequence you search in the database is say 0.971, do you want the algorithm to stop and report the result, or do you want it to keep searching and tell you if it finds something at 99 or 100? All algorithms deal with that differently, and you need to tell it when to stop or not. Another important consideration is, do you want it to tell you if there's more than one hit at the same ID, and which one do you want it to choose? So if it finds 2 things at 99%, then which one do you want the algorithm to tell you is the answer? If you need the results to be deterministic, then you need to tell the algorithm to choose the same one every time. Otherwise, there'll be some stochasticity in the process. So in summary, we've produced our table now. We were able to fill in the column labels immediately after experimental design, the row labels after classification or annotation, in this case denoising, because our table is representing denoised sequences, and we're able to fill the numbers in after mapping. The last thing that we might want to add to the table is some classification annotation on the sequences. So in this example here, our organisms here are represented by an ASV number or an actual sequence. And so we might want to add some taxonomy to that. So computationally assigning taxonomy is kind of easy, although, or I should say getting an algorithm to annotate sequences is kind of easy. In reality, classification is really, really difficult, and maintaining a database that's well annotated is also very difficult. And therefore, anything coming out of a classifier needs some kind of manual curation, I would say. So you should know what type of classification you require before you commence your study. So if you require just general bacterial classification against everything in the soil, you can just run against the database, and you know, you probably don't need that much information, and you're probably not going to get down to a low level. But if you need identification of a specific strain that's potentially pathogenic, then your requirements are much more stringent, and you need to choose your amplicon and the database much more carefully to negate false positives and false negatives. You also need to ensure that the target region has the required information. So for example, type 1 and type 4 Synechococcus ecotypes, and these ecotypes are the most abundant organisms in the ocean. They're differentiated by a single base pair and 16S. So they're differentiated by more than that in their whole genomes, but in 16S, there's only a single base pair that differentiates them. And that's seen in the first few hundred base pairs of 16S genes. So if you sequence V4 or V6, those things are going to be identical. You're not going to be able to differentiate them. And if you need to differentiate them for your study, then you need to sequence the V1 or V2 region. Bradyrhizobium species are also difficult to resolve at the 16S region. And so if you're interested in resolving Bradyrhizobium species, then you need to choose the appropriate variable region in 16S to be able to do that. You also need to consider termination options. Again, this sequence classification is essentially a mapping exercise against a database, and you need to understand which database you should use. So just to demonstrate the importance of choosing the database and what's in the database, I've kind of got this trivial example here where I'm starting with this lemon, and I'm essentially, when I try and classify against the database, I'm not really asking what have I got or what is this. I'm asking, is this, so I've got two, sorry, I should have said I've got two databases here. So one represented by the apple and the orange, and one represented by the apple and the orange and the lemon. And in the first example, I'm not asking what is this lemon or what is this. I'm asking, is this an apple or an orange? And the algorithm's going to say, it's an apple, it's an orange, or I don't know. And so if I set a 97% threshold and I say, is this lemon an apple or an orange? In this case, it's 95% similar to an apple and it's 97% similar to an orange. So the algorithm's going to tell me it's an orange. All right, and now you have to think that you're dealing with something unknown here. And so you're not really asking the algorithm, what is this, when you compare it to a database. You're asking, of these things in this database, what is this? So so in the first case, yeah, is this lemon an apple or an orange? In the second case, we can say, is this lemon an apple, an orange, or a lemon? And we can see that that we do have a match to a lemon at 100%. But if we tell the algorithm that we're happy with a 97% match, and we tell it to terminate when it finds a 97% match, it's going to go, is it an apple? 95% match? No. Is it an orange? 97% match? Oh, that meets our criteria. It has to be 97%. So let's call it an orange. So if you terminate the search too fast, you're not going to find out it's a lemon. So these are all things that need to be considered in doing the search. One way we can terminate the search more quickly is to change the order of the database to have lemon represented first. And there are various ways to do that and various algorithms that work in that way, but we still don't want to terminate the search too fast. I'll illustrate this point one more way. So, and this is important from a QC point of view. So in this example, we have a whole bunch of archaeal reads, and what we're trying to do is classify them or work out which ones are archaeal. We could classify them against the full silver database, and silver includes Archaeobacteria and eukaryotes. Or because we want to limit the search space and make our search faster, we could just pull the archaeal reads out of the silver database and match them against the archaeal reads only. And so this is a real world example here where I took a data set of archaeal reads, and if I search those reads or try and classify those reads against just the archaeal data from silver, 100% of them come back as archaeal, because I'm asking the algorithm, which of these things does my sequence match? So which of these archaea does my sequence match? And 100% of them get returned as likely archaea. But if I match against the full silver database, so if I include the bacteria and the eukaryotes in that search as well, and I say, which of the things are these sequences? It turns out that only 85% of them are really archaea. And so if you're trying to work out which things hit target and to try and get rid of contamination, then you need to give the algorithm the chance to choose something other than what you think the organism should be. And so it's really important to have a broad database and to understand what's in the database. So in terms of sequence classification, you need to know your requirements, and you need to choose a method that enables meeting those requirements, and think about the implications of false negatives versus false positives, and know the database and its limitations. And so with that, we've covered experimental design, wet lab issues, touched on sequencing, and had a really quick run through a basic bioinformatic workflow. And we'll stop there because I've already run quite a bit over time, sorry. And I guess next time we can deal with statistics and data stuff.
Yes. Yeah. Thank you, Andrew. I think we have more components to take, and maybe we can take tomorrow. Or how do we go about it? Yeah. Am I audible?
You are audible, Annapurna. Yes, whatever you can do, Andrew will take today.
Yeah,
and any more we cannot do tomorrow.
Exactly.
We can take the questions for what he has taken right now. There are 3-4 hands which have been raised. Kumar, I think the first 3 hands which have been raised, we will take the questions.
Yeah, there are too many questions, ma'am.
And the rest of the thing, Andrew, we can take tomorrow, the data storage and okay. So the first one is Chauhan. I think you can yeah, Navneet, you can ask your question. Or I think he's unable to. So Kumar, you can sum up and then we can take the questions.
Kamalesh is online. Kamalesh Jangit is online. Yeah, you can ask your question, sir. You put a couple of questions on the screen. You can ask directly to Andrew.
You have to.
Kamalesh, please.
You have to give the accession to him first. I think you should read out the questions, Kumar.
Okay, we can read out the questions. Andrew, can I read out, ma'am?
Yes, Kumar.
Yeah. Andrew, it was a wonderful and excellent talk about the metagenome analysis. I think you touched the very basics of this analysis. I'm sure it must be useful to all the students. Those who missed it, they can see YouTube also. The question here is from very important questions from Kamalesh. Is normalization for sequence number still necessary? What is your recommendation? We still do it because we have practically seen difference in output with either approach.
Yeah, can you hear me still?
Yeah.
Yeah. It's kind of depends on the question a little bit and what you mean by normalization. I think for alpha diversity analyses, you need to normalize the data somehow. I mean, it's really clear that if you sequence deeper, you'll end up with more diversity. So you need to control the sampling effort, unless of course you're in if you do coverage curves and you've saturated the coverage curves, then you probably don't need to, but I mean, that's pretty uncommon. So I'd say you need to normalize for alpha diversity analyses all the time. And then there's different schools of thought on that for differential abundance type testing. But essentially, if you're using some of the more modern, I guess, newly popular methods around compositional data analyses. So some of Greg Glaw's CODA methods, for example, essentially the read abundance is a probability, and through this center weighted log transform, that normalization is taken care of. So so up front you don't need to, although that is taken care of to a certain extent. So in a beta diversity / differential abundance, I think that those CODA methods are probably the safest ones to go to. But I think for alpha diversity, you definitely do need to normalize. Although, no, for alpha diversity, that's true. I will say with the beta diversity stuff that although conceptually it's clear that you need to deal with that stuff, in a real world situation, the patterns are really driven by the dominant organisms, and it doesn't really matter what you do to the rest stuff. It doesn't affect those beta diversity patterns all generally. But to be on the safe side and keep reviewers happy, deal with it.
Yeah. Another question from the same participant. We have seen clear difference in accuracy of assignments with the Silva as compared to other databases like Green Genes. Is it true, or is it really this observation is correct from his part? What he sees, Silva is having a much better annotation as compared to others. Yeah.
Silva has a more complete annotation. I mean, whether you trust that the guy is annotating Silva or not, there's a lot of errors in Silva, like there is in all databases. I would recommend not using Green Genes because it's deprecated, so it hasn't been updated since 2013, I guess. So the genome tree database is a more current version of that taxonomy from Donovan Parks and Phil Hugenholtz. The genome tree database taxonomy has actually been adopted by Silva more recently. Yeah, and then it kind of depends on the implementation of those databases that you use as well. So if you implement Silva through the QIIME architecture, for example, they include annotations down to species level, but if you implement it through the mother architecture, patch loss doesn't include any designations below genus, although they do exist in Silva. In the mother version of Silva, they're not there. And so, I mean, it's kind of, it really depends on the region you're sequencing in and the organism that you're looking at. But I mean, the chances of really getting reliably to species from a short 16S or short ITS read, I mean, we could argue about it, but I'd be dubious about your ability to really do that. But yeah.
Yeah, another question is how much percentage of non-microbial reads are considered as quality reads? Is it 20% or 10%? When you go for the total reads, certain percentage of reads are not specific for microbes. So what is the cutoff to consider the entire reads are high quality reads?
I'm not sure I quite understand the question. I would say that if you're sequencing bacteria with a bacterial specific primer, then anything that's not called bacteria, you should remove. It might be real. I mean, there's no doubt that if you're sequencing a fish gut and you get salmon read back, that it's a real sequence, but you're not targeting it, and so you're not getting getting a broad breadth of all of the reads anyway. You're targeting bacteria, so you should remove the bacteria. How many? I mean, it's kind of a funny question. So essentially, you need the depth of the bacteria to be enough for you to answer the question. So if you've got 50% like 50% host reads, then and you need 20,000 bacterial reads to answer your question per sample, then you need 40,000 reads per sample. I mean, or you need to get rid of the host some other way. Yeah. Maybe Paul, did you have something to say there, Paul?
Yeah, assuming I can be heard.
Yes, sir, please.
Yeah, you can.
So one of the examples that Gupta had up was shoots and leaves and adjacent soil and roots. And there, the contamination we had was that, you remember that mitochondria and chloroplasts are basically bacteria as well. They've been host resident for a very long time, but they're effectively bacteria. So the primers we used for that study picked up the wheat mitochondria and chloroplasts. So they just appeared as rows in there, and we pulled them out afterwards. So although they were specific primers, those primers worked on bits of the wheat plant as well as on the bacterial genes that were present.
Yeah, so not all.
Yeah, so you wouldn't normally expect, if you've got a good primer, to pick up bits of wheat, but you might pick up bits of bacteria embedded in the wheat, so to speak.
Yeah, but yeah, and not all primers do. So that 27519 isn't pick up so many wheat, I guess. And there are plastid minus primers, so primers that that don't target or that omit chloroplast reads more readily. And often it's also possible to size select host mitochondrial DNA out or host mitochondrial fragments out. They're often longer, so you can run a gel and size fractionate those out. Yeah. Not always, but often, yeah.
And you also talked about doing a QC step where you look at the size of the merged amplicons, and often that'll help get rid of some of that off-target stuff as well.
Yeah.
Yes, sir.
Yeah, Dr. Andrew, it's a nice presentation. You gave an overview about the strength and weakness of the whole exercise, which we do with the sequencing. I have a question. How do we add or account for the extraction efficiency, or PCR, or DNA extraction efficiency in our whole exercise, so that we can, improve the, what you call, process rate measurements or nutrient flux measurements based on the sequence data or quantitative sequence data?
I don't know that quantitative sequence data is a difficult thing. There have been various attempts to improve the quantitative nature of sequence data, so you can spike in controls at known concentrations. So Noah Fear has written at least one paper on using spiking controls. The difficulty is that to do it routinely in samples that you don't know is really difficult, because if you spike too much in, you swamp the signal of everything else. And if you don't spike enough in, you don't pick the signal up. And so if you've got an experimental system that you understand well, it's kind of a good thing to do. There is a group from Japan that have developed a whole bunch of synthetic oligos to spike in. So that's the other thing. You've got to choose something that's not in your sample to spike in, which is, depending on your system, not necessarily that easy to find, but there are a whole bunch of synthetic oligos that have been developed but haven't been that widely used. do it. Another way to do it is to standardize to total bacterial count. So you can do, for example, 16 SQ-PCR to get an estimate of copy number at least, and an estimate of biomass. And then you can standardize the amplicon number to that per sample or normalize against that. In terms of DNA extraction efficiency, it's a kind of tricky one. Generally, if you're doing a broad survey, people just use a commonly employed method. So most commonly the MoBio, or what was the MoBio kit. Who QIAGEN have it now, I guess, do they? Yeah. But if you're interested in a specific organism, then you need to make sure that the method that you're using will disrupt and free the DNA from that organism. So if you're interested specifically in spore formers, for example, then you need to make sure you're using a reasonably robust method to get that DNA out. And that just comes down to optimization using those organisms. It's a really tricky one. One thing that we could do more generally as a community, but we don't because it's expensive, is to use 3 or 4 or 2 different methods on every sample and then pull the DNA from those methods so that we are getting a broader suite of DNA. And for the same reason, we could also amplify the amplicon with different primers that target a similar enough region so that we can align them, but the primer bias is less because we're using different primers. But generally we don't do it because it's another library prep and it doubles the cost or triples the cost if we're doing 3. So we generally talk about DNA extraction bias and PCR primer bias, but we kind of ignore them. So yeah.
And add on to this, and how do you differentiate total bacteria from active bacteria? I mean, active bacteria, bacteria.
I don't think you can in a sequencing sense. Some people would argue that you can do an RNA extraction and then sequence the rRNA instead of the rRNA gene. But given that 16S RNA isn't transcribed and it's a structural protein and it's quite stable, I mean, it's a pretty hard sell to say that those transcripts are really representing actively metabolizing cells. I guess if you're targeting a more functional gene and you're targeting mRNA transcripts, you could argue that the organism is actively transcribing the gene. Whether or not it's being turned into protein and is active is a different story, but you could argue that it's active in that sense. So short of live dead type staining and kind of microscopy methods or flow cytometry methods and some kind of in situ hybridization at the same time, it's difficult to prove absolute activity. You can measure the activity, so you can measure denitrification, but to show which organisms are actually doing it is like a really tricky thing. Yeah. I guess the other thing I should say is that for anabolic processes, stable isotope labeling is one method. So if something's fixing carbon, for example, you can label the system with a heavy carbon isotope and sequence the DNA from that. And there's also work being done with different isotopes around water and phosphorus to look at things incorporated into DNA and activity generally rather than associated with a certain process. So you can do it that way, but it's kind of tricky.
Thank you.
There's a question from Shantanu from School of Environmental Studies, Jadavpur University. He asks, how can I perform network analysis of 16S OTUs under organic and inorganic fertilizing practices? And is there any effect of heavy metals on bacterial community composition and abundances?
I guess in answer to the first question around network analysis, there's a whole bunch of of tools available online, and there's a whole lot of self-learning resources associated with those tools. So SparCC would be one. I think the FileSeek package has network analysis embedded in it. There's also the Mine package, and there's an online tutorial. It's kind of a bit older now, but conceptually it's still the same, from Jed Furman's group, looking at Pearson correlations, and then essentially reading those into Cytoscape and doing those kind of analyses. There's also a review on network analyses and identifying network keystones. I'll call them network keystones rather than environmental or ecosystem keystones. But so Sam Banerjee wrote a review in I think maybe in Nature Reviews Microbiology, with a whole bunch of methods on that. So they'd be good places to start. Yeah, metals do have an effect on organisms. Difficult thing is to work out whether the metals are available or not, or whether they're not available to the organisms, and they're bound to the environment. So you might be able to see the effects of those metals through different resistance genes, for example, or I guess if you're interested in gold and the effect of gold and the cycling of gold on microbial communities, you could check out the work of Frank Reith from University of Adelaide. Yeah.
There's one student, Navneet Raj Singh Chauhan. He can unmute and ask the question himself. Navneet, can you hear me? He has raised his hand. Praveen Rahi.
Yes. Hi, Andrew. Thank you, Dr. Annapurna, for giving me the opportunity to ask the question. Yeah, so my question is very direct, Andrew, that if we have a data generated based on 215:2 chemistry, can we still get good merging for V3 and V4 region? Because my data is generated in 2 into 250 chemistry, and I'm losing a lot of data at merging step. Like 98% of my data is losing. So
yeah, so it kind of depends how long your fragment is and the quality, and it depends a bit on what merging tool you're using. So I'm
using this KIND2 pipeline with Data2.
Yeah, so
the recent one.
Yeah, so you might try a tool that doesn't. So Data2 does the denoising before it merges, and so if it's trimming a lot of your read 2 because of its low quality, then you might be trimming too much. So you could try another tool that doesn't require that merges before the denoising step. So I think the mother merger or the USearch merge tool would do that. Or actually, a merge tool that definitely does that is called Flash.
Okay.
Flash 2.
Yeah, Flash is very old tool. People are using it previously also. Yeah.
Flash is really fast and it's pretty good, and it's pretty yeah. So the problem with one of the things with Mother is that it will concatenate unmerged reads, so doesn't dump them. So you have to be careful to remove them. And the USearch tool by default only accepts like I think one mismatch, or like not many mismatches. So you need to as the default. It's a specified number, so it doesn't scale to the length of the overlap. So if you're overlapping 100 base pairs, you would expect a different number of mismatches than if you're overlapping 10, for example. Yeah. And the default in the USearch tool is a hard number. So, but you can change that and tell it a percentage. So you just need to play with the arguments and choose something appropriate. But out of the box, Flash is kind of okay, or like it works pretty well. The other thing that you should do is try and merge the sequences by hand. So just open the FASTA files and pull 2 sequences out and align them. And so if you can align them by hand and the algorithm can't, then you know that that you should be able to change the parameters in the algorithm to get it to do it. If you can't merge them, if you can't merge them by hand, you know that there's a problem with the sequencing step. Yep. So looking at the data is a good way to go.
Yeah. Thank you.
Okay, there's one question, hand raised, which is Galaxy something. If you want, you can ask by unmuting yourself. Galaxy.
And there is another straight question. What's the difference between OTU and ASV?
You could argue nothing. An ASV is an amplicon sequence variant. OTU stands for operational taxonomic unit. So your operational taxonomic unit could be the ASV, but more typically it is defined, and your operational taxonomic unit could also be genus or family, but typically in a molecular sense, an operational taxonomic unit is defined at a set sequence similarity. So 97% sequence similarity or 99% sequence similarity. And so it's a hard cutoff, but yeah, so an ASV is an amplicon sequence variant, which is essentially a denoised sequence. And it is exactly the same as an exact sequence variant is another term. And the other term that's commonly used is a ZOTU, which is a 0 radius OTU. And they're all the same.
There's one more from Siddharth. I think Siddharth Shankar, you have raised your hand, you can ask your question. Unmute yourself and ask your question.
चल रहा है
Siddharth.
लास्ट टाइम यहाँ पे सही थी बट वहाँ पे बदल जाती है
There, Siddharth? Then we ask Kedar. He's also raised his hand. He can ask his question. Kedar?
Yeah, good afternoon, sir. Good afternoon, madam. My question is how to effectively choose the data that are randomizedly not perishable.
Can you
repeat the question?
Sir, how can we collect a data which is, hello?
Yeah, please.
Sir, how can we collect a data without losing its randomity and uniformity, sir?
I'm not sure I understand the question.
How
on the question a little bit?
The data without losing its uniformity and randomity.
Yeah, I heard it. I just don't understand it. Sorry.
Okay.
Can you explain a little bit more, or?
Yeah, probably he means consistency in the results, sequence data between samples, within samples.
Yes, sir. Yes, sir.
Reproducibility of the results. How can you maintain that?
Consistency, reproducibility. Yes.
Okay, so well, so I guess this comes back to understanding the variability in the data, and essentially there's variability produced in each step of the process. So if you extract the same sample, extraction is a destructive process, but if you were to extract the same sample twice, you would end up with a slightly different answer. If you were to PCR the same aliquot or the same extraction more than once, you will end up with a slightly different answer. If you produce a sequencing library from each of those aliquots, you'll end up with a slightly different answer. If you sequence the same sequencing library on a different sequencing run, you will end up with a slightly different answer. And that was really apparent in the mock community example I gave, where all of those bars looked a little bit different, and they are all the same factory produced certified sample. So they should be exactly the same. The thing that you really need to make sure of is that the estimate that you're getting of the of your sample is a reasonable estimate of the mean. And so if the sample that you've got is highly variable, then you might need to perform the step where that variability is coming in a couple of times. So if you've got a kilo of soil and the variability is coming in the DNA extraction technique, you might need to do 10 extractions and pull the result so that you're getting a reasonable estimate of the mean of that thing. But and we're never going to get rid of all of that variability. What we're really trying to do is to make sure that variability is less than the variability between samples. So the data I showed you on those mock communities, they all look different. And if we analyze them or we compare them to one another, they're all different. But if we actually compare them to one another in the context of all of the other samples on those 15 sequencing runs, they look identical. They're so close to one another that you can tell that they're all that mock community. It's just that when you only compare these two things, they appear different because we're concentrating on the differences in those two things. So understanding your system and trying to get a handle on where the variability comes and trying to make sure that you're controlling that is the important thing.
Thank you. I think, Kedar, you can ask your question. Can you hear?
Madam, another question. It's again a straight question. What exactly is the difference between poster copy and genome copy? Are they related to each other?
Yeah. So in that example, sorry, I didn't probably explain that very well. The poster copy was the number of 16S copies that the manufacturer reported in their documentation, and their documentation was a poster, like a scientific poster. And so there was a table on that poster, and that's where I took that data from. The genome copy number was the number of 16S copies that I found in the genome. So I downloaded the genomes, and I used a tool to pull 16S genes out, and I found that the number in that column, and those two numbers weren't congruent. They were different numbers. Interestingly, the number that I found by using their genomes was the same as the numbers in the annotations of the genomes when the annotations were available. So I trusted my numbers more than their poster numbers, but the poster numbers are what ship with the mock community. So if you just trust the documentation that ships with the community, then it's not correct. Although what difference it really makes if it's 14 or 15, I mean, probably doesn't make any in the real world, but it's just an example to show that sometimes it's difficult to assess these things. Yeah.
Thank you, Andrew. One last time I'm asking Kedar. He's again raised his hand. Are you there, Kedar? Okay. So I think
we can carry forward to tomorrow also, ma'am.
Tomorrow. So we are just about near 2 p.m. here as agreed upon. And thank you very much, Andrew, for such an excellent talk. And we'll take forward tomorrow again. And
10 o'clock.
10 o'clock in the morning for us. And I don't know what time it is yours.
2 p.m.
2:30. Nice talk. Great talk and very informative.
2 p.m. Both were excellent talks. Thank you so much, Gupta and Andrew. And we'll meet you tomorrow. Same time.
Yeah. Thank you very much.
Excellent. Thank you.
Bye.
Bye.